So good morning, everyone. Welcome to the 21st edition of our International Symposium, Priorities of Chemistry for a Sustainable Development. It's a great pleasure for us to meet you here all, and also our online participants. Before starting the event, I would like to thank our sponsors, Metrom, MDPI, New Green Protocol, Nano Team, FANOS, as well as our exhibitors, Amex, our publication partners, Frontiers in Materials and Polymers, Pharmacia and Emerg. We should say that this event is organized under the aegis and with the support of the Romanian Ministry of Education and Research through the National Authority for Research, and in collaboration with the Romanian Chemical Society, General Director Foundation, the Association for Development Education, Integrative Education for the Future, and with the support of our media partner, the Radio Romania Cultural Field. And this edition is for the first time held under the aegis of the Academy of Romanian Scientists. It's a very important aspect for us also. I will try not to be too long because we are a little bit in delay to the schedule. It's not a very good sign to start with a delay. So for the opening words, I will invite our General Manager, Dr. Michaela Arduini, please.
Thank you very much. We send to you a very warm welcome to our PRIOCHEM 21st edition. We are happy to see here old friends, and also we see new, hopefully, friends here. We hope that at the end of our symposium to establish new collaboration, to learn new things, and to try to move forward with our research results. We thank you very much because you are here. In this period, there are a lot of events, scientific events, and I know that it's difficult to choose where to go, but we thank you very much that you decided to be with us these days. I wish you very productive discussions, and to find during this presentation, to find new things and to learn and to maybe to have new ideas of research. Good luck, and I hope to see you also in the end of our event, because we have a lot of prizes, and we hope to be able to give to you these prizes. Good luck. Thank you.
Thank you very much. As I previously mentioned, one of the co-organizers of this event is the Romanian Chemical Society. So I will invite Professor Michaela Dina Stănescu to say a few words. Also, we have a presentation here, so I will ask my colleagues to run it. Professor Stănescu, please.
Yes, I share the presentation, or you put it on the screen?
We will put it on the screen.
Okay, I am looking for.
Yeah, it seems that we have some small issues. So Professor Stănescu, if you could run it yourself, it would be great for us.
I will share. I put share. You allow me to share it, to share my screen?
Yes, you should click share.
Yes, I click share, but it's totally another thing. That enter. And just a minute.
Yes, we can see your screen.
Because there are a lot of things on my screen. So now.
It's perfect, yes.
Okay, and I will. Okay. So first of all, please remark that we have a new design for the periodic table. This is performed by UCHEMS. which is a European society of old chemists, and it shows beside the element also the quantity you have it and the risk to not find it later on if you use too much. So it's very interesting, and we'll put it on the site of our society also. Why chemistry and chemical engineering? It's a question, but the answer is very easy because chemistry has a lot of application, and some of the field are shown here in this slide. But now we have to think differently in doing work in this field. We have to think by the goals of the sustainable development, the 17 goals, which I am sure everybody are knowing today. Why so? Because we have to think of the future. We have to think it's a future generation. We have not to use everything that it is now and like in the saying carpe diem, because we have to think at tomorrow. And for that, we have to change our way of developing industry, science, everything by innovation for pollution removal. It's a climate change that it's tremendous in the last days. Also, we have to make it green energy. We have to think about circularity. I just had attend Euroactive workshop about circularity. It seems that in the world it's 6% of things are just put in and reused, and in Europe 12%. So it's very, very little. So the waste valorization, it's just an important target. And also bioeconomy, it's the future, because bio-waste can be recovered, and we have to valorize it and apply the biotechnology, because working with enzyme, we have a smaller consume of energy, of also of reactive and so on, and less, of course, less pollution. And so we have to develop a new policy and a new approach concerning our personal training and way of seeing things. We have to improve continuously our knowledge. We have to think always when we develop some research of the environment, of preserving biodiversity, of using regenerated raw material, of protecting food safety and security, and also of embracing the interdisciplinarity and teamwork. Nowadays, you can't solve a problem knowing only one field. You have to have a team composed by people having knowledge in different fields and so. And talking about teams, I will say some words about Romanian Chemical Society. We have now around 1,200 members. They are just in territorial, organized in territorial branches, and usually they are organized around the university. And also we have professional division. We are just now trying to reorganize for making it more efficient, because the idea of making this division is to put people having the same interest together for communicate, for making teams, for making project and so on. And I do hope this reorganization will just lead to such future for the division, because how they had have worked till now wasn't very satisfactory. And also we have a publication, our special journal, and I just announced the interested people that on the site soon you will see the second issue of this journal, which will appear very, very soon. It's already, we have to put it on the site. And last but not least, I wish you a very interesting experience. Really, such a meeting has a purpose for making communication, for starting a new collaboration, and so on. Also, I wish you a successful presentation, and it's very important to know that it's not the prize that it's important, but it's a participation, because you just will have a new experience, new friend, and so on. And last but not least, I just share with you my, let's say, think about how the researcher may be. It has to have to foresee seeing, to have seeing an egg and see the marvelous birds that will appear after that. So good luck for the meeting, and thank you for letting me talk and say something about the Romanian Chemical Society. Okay?
Thank you very much. It is always a pleasure to hear you, Professor Stănescu. Thank you very much for your message.
Thank you also.
With your accept, of course, I will now invite the president of the Romanian Society of Chemical Engineering to say a few words to us.
So, it's a pleasure for me to be here again. If it's interesting for my speech, very short, I use Romanian language. So, eu cunosc ICECHIM-ul de când eram copil, de când eram student, în 1970. Deci eu cunosc doamna profesoară ICECHIM-ul de când eram student. În 1972 am făcut practică de vară la ICECHIM, unde am învățat cum se face un colorant pentru colorarea PVC-ului, pentru că se căuta să se facă așa cizme drăguțe din PVC care... Bun, a trecut foarte mult timp. În 1995, după 1990, ICECHIM a intrat într-o perioadă neagră. Noi, un grup de oameni, am vrut să facem Societatea de Inginerie Chimică. Și ne-am gândit, și între membrii fondatori ai Societății de Inginerie Chimică, ICECHIM-ul este unul pe care noi l-am ales atunci când aveau mari probleme. Timpul a făcut ca să se rezolve totul. N-am pierdut nicio participare la manifestările PRIOCHEM. Am ratat acum să mă înscriu cu o lucrare, pentru că noi organizăm prea multe manifestări în țara asta. Noi pe 6 și 7 avem manifestarea ICECHIM 2025, o manifestare destul de interesantă, unde toată lumea este așteptată. Mai putem încă înscrie lucrări. Dar, deci, ICECHIM organizează PRIOCHEM. Noi organizăm SICHEM. Facultatea organizează RICE. Se mai organizează. E momentul și e timpul să ne unim, să facem manifestări mari, manifestări de 500-1000 de participanți cu lucruri deosebite și să venim să ne cunoască lumea. A doua problemă pe care trebuie să o spun, noi suntem un grup de oameni care facem inginerie chimică, chimie, ce doriți dumneavoastră, trebuie să reușim să ridicăm o revistă națională la standul de revistă cu acreditare IC Thomson. Trebuie să ne unim. Am avut revista de chimie care a murit și moartă va rămâne, așa că, mă rog, și moartă va rămâne, așa că e cazul, societatea de chimie, societatea de inginerie chimică, societatea de biotehnologii, există o grămadă de societăți, da? Și mai ales institutele de cercetare, da? De cercetare trebuie să aibă, domnule, să publice, să fie acolo, IC 3, 4. Aș putea să vorbesc mult, dar mă opresc aici și vă mulțumesc mult pentru atenție. Încă o dată, Societatea de Inginerie Chimică, fondată în 1995, este membru a Federației Europene de Inginerie Chimică și noi suntem, prin Federația Europeană de Inginerie Chimică, prezenți acolo, la Bruxelles, unde se mai discută și despre probleme de cercetare, probleme de proiectare, probleme de orientare, de dezvoltare inginerească la nivelul comunității europene. Deci, suntem acolo, am fost și eu odată în polurile alea pe acolo, era să mă și pierd. Vă mulțumesc mult pentru atenție.
Thank you very much, Professor Dobre. I have to announce, it is a pre-announcement, let's say. Revista de Chimie will be or managed and organized by ICECHIM in the next future. Yes. So, I think that is a good news for our field in chemistry and chemical engineering. And of course, we hope to have your help for all the societies in chemistry and chemical engineering. Thank you.
Thank you. I will invite our scientific manager to say a few words, please.
Thank you. As usually, in the middle of October, it's a great pleasure for us to host this scientific event, which is based on interdisciplinarity and its focus to application. I hope that we'll pass the difficulty due to the underfunding from this period, and we will continue to develop our scientific approach and to have a result with great impact. I wish you successful meeting. Thank you.
Thank you very much. As we previously mentioned, the traditional partner, Romanian Chemical Society, has a very important representative also in ICECHIM, Professor Andrei Sârbu. It's the head of branch 3 of the Romanian Chemical Society. So, Professor Sârbu, if you would like to say a few words, please.
Hello, everybody. I am very happy to meet you again in our important symposium, I think. It's a traditional already symposium, well appreciated in country. And this is because every year we try to be better. And I think that we succeed to make better and better our symposium, and this attracted new participants, and we are very happy that we succeed to unify the interest of different branches of chemistry and in chemical engineering. I am very glad to see here many young people. Our aim is to put a very important point to focus on young people, because they are the future of the chemistry. And I wish success to this symposium and to have nice hours spent here, and to succeed to obtain new cooperation, new idea for the future. Good luck.
Thank you very much. We have several guests also present today in the room. So I will invite Professor Roxana Rădvan, the General Manager of one of our traditional partners, INOE 2000. So Professor, please.
Thank you so much. I like to add also that I'm extremely proud to be here, that I represent a large collective of collaborators of ICECHIM with a long tradition, and it's really a must to say that I'm not only honored like all of us here, but I like to confess also that I had a lot to learn from the good partnership we have, from the hard worker teams which I always met in ICECHIM, and from a special strong leadership and the devotement which I saw here in this institute. In this context, I'm very happy to give an award for our guests and organizers from our institute, also for your special anniversary. 75 years is not a common moment.
Thank you very much. You are full of surprises. Thank you. In fact, we celebrate this year 75 year. We are, I think, young.
From the beginning
yes.
for such work, indeed.
Yes. And so thank you very much. Indeed, we have a very nice collaboration, and I hope to be a stronger collaboration in the future, and to have a very nice and leader partnership, let's say, not only in Romania, but to have European collaboration very strong. And yes, and so thank you very much.
Good luck and a successful event.
Thank you.
From our colleagues as well.
Thank you very much. Of course, the floor is open. If any of our guests would like to say anything to us today, it's perfect. Okay, then I will pass the microphone to Dr. Michaela Dini to chair, basically to introduce our next speaker, because we have a single plenary session section, and after that we'll have a short break. To all our colleagues in the online, I know we have some issues with connecting to the Zoom meetings. We will try to fix it during the break.
Thank you very much. So we will open now the first plenary session. I hope that Fabiana is online. So I will introduce the first plenary speaker, Professor Fabiana Arduini. I want to say that I met Fabiana more than 20 years ago in University Tor Vergata in the laboratory of biosensors. Then she was a very young and promising PhD student, and now she is a brilliant professor and researcher in the same university. So I'm very proud that she accepted to open our plenary sessions. Fabiana is now full professor at the Department of Chemical Science and Technologies in University of Rome Tor Vergata. She is founder of the startup Sense4Med and the director of quality assurance at the ISO 9001 certified laboratory lab cup, also in University of Rome Tor Vergata. Fabiana works in the research field of miniaturized electrochemical devices, mainly using screen-printed electrodes modified with nanomaterials and paper-based analytical tools, applied in biomedical, agri-food, and defense sectors. She has published more than 200 articles in peer-reviewed journals with an Hirsch index of 62, and she co-authored more than 10 patents. She is the coordinator of several national and international projects, including recently Horizon Europe Pathfinder project with acronym Phoenix OOC, and for this, her name is listed in the top two of most cited researchers in the world, 2%. Also, for all this achievement, she is the editor of Green Analytical Chemistry Journal of Elsevier, associated editor of Microchemical Journal Elsevier, a specialty chief editor for micro and nano sensors and in frontiers in sensors, and so on. We thank you very much, Fabiana, for your kind acceptance to open our plenary session. Please, I hope that you heard us, and you can now start with your presentation.
So before to start, I would really thanks to Michaela for the invitation and for very kind presentation. So I would like to share my screen. Please let me know if you are able to see my screen in order to be sure that everything works well. So are you able to see my screen? Perfect. So as said by Michaela, I'm full professor Department of Chemical Science Technologies, and my mainly activity is focused in the last among 10 years in the development of paper-based electrochemical biosensor as sustainable analytical tools. So in this presentation, I would like to show you the journey in the last years in which we start with demonstration that paper is not only a sustainable substrate to produce a smart, environmental friendly electrochemical sensor, but can confer additional feature to the electrochemical device. Indeed, by harnessing the porosity of the paper, we are able to load the reagents in the paper network in order to deliver reagent-free device. In addition, by also harnessing the foldability of the paper, we are able to develop a lab-on-chip on paper, able to treat the sample, to contain the reagents, and to make the measurement. But not only, because these devices are able also to break down some limitation of classical electrochemical sensor, like, for example, the capability to detect the target analyte not only in liquid phase, but also in aerosol sample without any additional sampling system. In addition, these devices are able also to help other technologies to solve some issue. So work well also in hybrid system. In addition, I would like to show you what we are doing now in my group. For example, the development of paper-based electrochemical device assisted by artificial intelligence for a smart detection. Or also we want to break down another pillar, like the use of paper for the advanced smart organ chip device. In addition, just a few words regarding the possibility, what we are doing now in the Sense for Med company, the possibility to use this device in the for market entry. So just I would like to show you the starting point. So we have experience in the development and production, mass production of screen printed electrode. So we start the use of polyester as support, And in addition, in order to improve electrochemical performance, to maintain the sustainability vision, in the last years, we use the carbon black as the nanomaterial to improve electrochemical performances of this device. We demonstrate the suitability of this nanomaterial in 2010, and after I can say that a lot of people around the world use this nanomaterial instead of more famous, for example, graphene and carbon nanotubes, because which is the advantages of this device, of this nanomaterial. So we are able to prepare a stable dispersion used for drop casting easily by only a sonication step. In addition, we can use as received by the supply, so without any additional treatment. And of course, is able to improve the electrochemical performance of the device in terms of low apply potential, increased sensitivity, resistance of fouling. In addition, the cost, 1 kilogram costs only €1. Reason for that, I said before, a lot of people now use this nanomaterial for production electrochemical sensor. This is the review that reported some example about this. So this is the starting point. So we are again, at the start, the use of nanomaterial or device to have a sustainable approach for the detection. How to render more sustainable this device? We started to replace easily the plastic support with the paper. And of course, when we use the paper, especially filter paper, and we have a drop of water on the paper, there is the diffusion of the water due to the capillarity of the paper. So it is characterized by microfluidics without any external pump. So it's good because it's easy to have a microfluidics. In addition, using paper, we are able to overcome the issue of the bubble. And of course, but we need to manage the fluid and to create hydrophobic region that delimitate the hydrophilic one. In this case, we use the wax printing to create hydrophobic region, so easily using the wax. And so we designed the pattern. So the green part is the wax. We printed the pattern, and by heating process, there is the percolation of the wax in the paper. And so after that, we have the microfluidic pattern in which we printed the electrode. So to produce the device, we combine screen printing technology to print the electrode and wax printing technology to have microfluidics. So I would like to start in my journey and to highlight, I said before, it's possible to use paper like a smart material for electrochemical device. At the start, I will show you an example, which is a biosensor that we developed in collaboration with Italian Ministry of Defense to detect botulinum toxin A and C. So actually, the reference method using the mouse and require 5 days to make the measurement. In order to have a more sustainable approach, our idea is to study how this toxin works in our body. So he's able, the different toxin, to cleavage some peptide. So our idea is to select a synthetic peptide in which are present the cleavage site of, for example, botulinum toxin A and C. So we can see that the red part is in which is present the cleavage site of botulinum toxin A, and this part is present the cleavage site of botulinum toxin C. So easily we synthesize this sequence, functionalize at the end with the thiol group cysteine in order to immobilize this sequence of paper-based electrode, in this case office paper-based electrode, functionalize with gold nanoparticles. And at the other part of this sequence, we modify with the methylene blue, which is an electrochemical probe. So what's happened in presence of botulinum toxin? So means that there is the cleavage, so it's possible to cut the sequence, and we remove the electrochemical probe close to the electrochemical surface. So means that in absence of botulinum toxin, we have the signal. In presence of botulinum toxin, there is the removal of electrochemical probe close to the electrochemical surface, so we have the reduction of the response. So easily, we have to ask to handle user only to add the sample, to wait the reaction, to wash, and to make the measurement. So we can see the calibration curve of botulinum toxin A, and we demonstrate that it's possible to detect a very low concentration of this toxin, this is the detection limit, and also the botulinum toxin C. And this is the paper, and also the Italian patent. And we are working in order to create an array in order to detect with a single drop of the sample the different botulinum toxin by mobilizing different type of the peptide. And in this case, which is the advantage of using, for example, paper? Of course, the low cost, but in the defense field, one pillar is the decontamination. Using paper is possible to incinerate the device after the measurement in order to decrease or totally eliminate the decontamination step. So demonstrating this, okay, so we are moving to demonstrate that it's possible also to use paper, not only as sustainable substrate as reported before, but also as a reservoir of reagents. This is an easy example in which we detect the essential oil within our Reliance project. In this case, we detect thymol, eugenol, and carvacrol, and we can see the peak. And the carbon black again, we observe an increase of sensitivity because the detection requires, for example, the use of acetate buffer. We preloaded the buffer salt on the paper using, for example, filter paper. So what's happened? We had, for example, 5 microliter of acetate buffer, and we wait that the solvent volatilize, and after the sensor is ready for the measurement, because we can take any sample, liquid sample, put the same volume on the paper, so we have the solution of the salt. We can have the reaction and the exact pH without any additional sample treatment, including, for example, pH adjustment. So another thing that I show you is possibility to have combine different features to have a lab on chip of paper. We demonstrate this, for example, in the case of a point-of-care device for the precision medicine in Alzheimer's disease. How work this biosensor? In this case, we have to detect the effect of some drugs, like, for example, riostigmine in the patients. These compounds are able to inhibit acetylcholinesterase enzyme, which is a key enzyme of neurotransmission. But this enzyme is present in the brain, but fortunately, we have also a butyrylcholinesterase enzyme in the blood, so it means also in capillary blood. So the idea is to evaluate the inhibition of the drugs by measuring the residual activity of butyrylcholinesterase in capillary blood. So we have how to carry out the measurement. So we have to treat the sample in order to have from capillary blood the serum. After that, we have to measure the residual enzymatic activity. So the enzyme is present in capillary blood. So means that we need of enzymatic substrate. So we use this pad to preload the enzymatic substrate. And after we have the measurement. In this case, we call this system like an origami. And in addition, we move. So the classical lateral fluidics, like in the classical point of care that we have, for example, in the pregnant test, so in vertical microfluidics. So with improving the time analysis because it's shorter and also the sensitivity. So we optimize the different pad. And we demonstrated that these drugs, like riostigmine, donepezil, also riostigmine, take as model, are able to inhibit both acetylcholinesterase, which is present in the brain, a butyrylchosterase is present in capillary blood. So means that the idea was correct. So we demonstrate the suitability of the device by measuring this target analyte at micromolar level. And the system is able also to work in venous blood, the capillary blood. And this is the device that we have developed. So this is the strip. This is the Sensit, which is commercially available, smartphone assisted. So really easily, we have created a lab on chip to allow from the patients by precision medicines in the case of this disease. So in addition, I said before, we demonstrated also we are able to break down some limitation like the detection in aerosol sample, target analyte, starting from this device, in which several years ago, we developed a system to assemble, to solder overall for detection of nerve agents in aerosol sample. In this case, we need to have this automatic system to sample there, put there in the solution to make the measurement. The idea was that we can use the humidity as actuator to make the measurement. How? Again, we can use an origami approach. In this case, we demonstrate this for mustard agents detection using choline oxidase as a biocomponent, because this enzyme is able to be inhibited by these chemical warfare agents. So again, to make the measurement, as reported before, we need to have the enzyme and the enzymatic substrate. So we preloaded both the enzyme and the enzymatic substrate in 2 different layers. So again, we use a porous paper, so filter paper. That after the addition of 5 microliter in this part of the enzyme, 5 microliter of this part of enzymatic substrate, we wait that the solution becomes dry. After that, we close each other and exposed this device to aerosol sample created of the mustard agents. Of course, not in my lab, but in collaboration with the Bundeswehr Institute of Pharmacology and Toxicology in Munich, Germany, and we demonstrate that it is able to detect the mustard agents in aerosol phase easily by putting the device in aerosol phase. So without this additional sampling system to make the analysis. After that, we can move also in the application this approach, for example, in breath analysis. And the first article was published by Professor Dinser, and in the same year in which we published the article of mustard agents. In this case, his group detected hydrogen peroxide in breath. Consider that, for example, the breath sensor for disease diagnosis was considered one of 10 emerging technologies in 2021. So we applied this approach, for example, for the detection of SARS-CoV-2 in breath, starting from this work in which we developed the first electrochemical biosensor during the pandemic event for SARS-CoV-2 detection in saliva. And using this approach, so this is again an electrochemical sensor in which we detect the spike protein or M protein by immobilize the electrochemical chain on magnetic beads, and again the electrochemical detection by using portable system. Use the same approach, but collecting the virus in the breath by using a paper-based device. So what's mean? We had these 3 pads in the mask, and we ask to the patients or healthy people to wear the mask and to maintain the mask for several minutes. After that, we take this reservoir, paper-based, and we make the immunological chain within this reservoir. So reservoir act as collect the exhaled breath sample and a site for incubation within the sample magnetic immunoprobe in order to detect the virus simply by wearing the mask. And we also evaluate the different position of the paper-based pad And we can see, and also all together, we can see that in all cases, we are able to detect the healthy patients and also the positive patients. Consider that the positive patients in that case was 31 as cycle threshold using real-time PCR, so means a high cycle, so very low viral change. So demonstrating again that our system is very, very sensitive. How we use this approach? We prefer to use the paper-based without chemical reagents in order also to have the possibility to have the regulatory approval easily, because if we preloaded the reagents in close contact to the body, to the person, after the regulatory aspect is not so easy. In that case, we have also paper-based pad in the breath that work very well for both applications. So after that, I will show you also to demonstrate the possibility that paper can also help other technologies to solve some issue. One is, for example, the possibility to use paper in combination with the crystallography method to have electrochemical device. In this case, we collaborate this without another European project, Serentex H2020, in collaboration with the Novi Sad University, Professor Goran Stojanovic. In that case, they supply this device, in which there is the PVC, two-layer close contact. In that case, we have a gold working electrode, cap, contra electrode, silver, and only one hole in order to contain the solution. So we can see it's not so easy modify, for example, the gold working electrode, because it's comprised by two-layer PVC. So how to create, for example, glucose biosensor easily? We use a strip of paper. So in the strip of paper, we preload the enzyme, also the electrochemical mediator, in this case, Prussian blue composite with carbon black. And this device is easy and ready to make the measurement. So by only add the strip, so preloaded with the reagents, and adding, for example, hydrogen peroxide, we are able to detect this target analyte. By adding glucose, we are able to detect the glucose in the sample, for example, in the sample tiers. So also in addition, thanks to the suggestion of the referee in that article, we preloaded also, for example, uricase. So using the same electrochemical mediator, the same strip, by adding, instead of glucose oxidase, uricase, you are able to detect uric acid, or by preloading, again, the nanocomposite constituted of carbon black and Prussian blue nanoparticle, and lactate oxidase, you are able to detect lactate. So it means that by easily changing the strip, this device is able to detect detect target analyte. Another example that to show you is possibility to use paper also in the wearable sensor in order to detect the target analyte is wet by using a very efficient and easy to use microfluidics. In that case, we detect cortisol using an immunological chain. In this case, is a competitive approach. So it means that we need the antibody to capture cortisol. In addition, we need to cortisol label with the enzyme. So how happened the measurement? In this case, when the person starts to produce wet, there is the wet of this part in which is preloaded the cortisol label with enzyme. So there is the flow of cortisol present in the sample of the sweat and the cortisol preloaded before label with the enzyme. After that, this sweat reach the working electrode in which is present magnetic beads functional to antibody. So we have the competition. And after the sweat flow again until the absorbent pad which work as a reservoir and passive pump. To make the measurement easily, we fold this part preloaded with enzymatic substrate, we make the measurement. And we combine this system also with a printed electronic device for NFC detection in order to have a flexible system for detection of cortisol. But of course, the limitation of the system is that we can detect cortisol one time, and after we have to replace the paper-based device. So because this microfluidic help also the detection in time, we demonstrate that it's possible also the continuous analysis of the target analyte. In this case, we select a target analyte in the pH and the sodium in sweat. And in that case, we construct this microfluidic, like a butterfly-like configuration, in which there is the sampling pad and also, again, the absorbent pad that work like a passive pump and a waste reservoir. So to make the measurement, we have only to add this device on the skin during the sportive activity. And the very important things to demonstrate that it's possible to detect, for example, the concentration of sodium exactly in that time without memory effect, we evaluate the hysteresis, and we can see that changing the concentration on the sodium in the sweat, we are able to detect the exact concentration without memory effect. We demonstrate this, for example, in the testing a different subject, and also at the end we collect the sweat and the measurement using the reference method. We can see the good agreement. Demonstrate that with this device we are able to detect the change of the sodium concentration in sweat, which is correlated to dehydration in the time without memory effect. This is the article. So just a few words about what we are doing now. Starting from this article, in which also we have a patent, we demonstrate that it's possible to detect the infection in orthopedic field, for example, during the surgery by developing this device for pH measurement. We also start to create a platform in order to detect exactly the infection in orthopedic field, for example, by due to the presence of peristatic joint in the body, by have an array of the device combined with artificial intelligence in order not only to evaluate the type of bacteria that produce the infection, but also the antibiotics to select. In addition, as at the start, we start also to use paper in organ chip for the development of organ chip device within a European project Phoenix, which is a pathfinder. We can see the different review about the advantages to use paper as smart support to develop electrochemical device. And in addition, in this recent review published in Electrochemic Acta, we demonstrate that paper, so paper-based electrochemical device, can be used for pharmaceutical industry control, also for drug residue in food and water surface, for precision medicine, in addition also for the development of the next generation organ chip for precision medicine. At the end, just a few words about what we are doing in my company. So I am involved in the two spin-off company. One is TechnoSense, which now these sensors for water molding are now in the market. And last year, TechnoSense was acquired by the holding Lasatec. And after the application of this, my expertise in water molding, I would like to push and moving from biomedical field. And we working to develop a paper-based device for the development of point of care for the sweat test analysis for the analysis of cystic fibrosis. It was like not so easy, just to say also crazy idea, because cystic fibrosis is a rare disease since more than 5 years ago. And just 2 years ago, the European Commission lighted that biosensor could help treat rare disease. So after, thanks also this highlight of European Commission, Sensor for Med received last year the first round of the funding in order that to produce the device, and we are now working to have a clinical trial in order to push our device in the market. So this is what I present in my journey. So I would like to say that this is one of articles that inspired me to start this journey in paper-based electrochemical device. And happy to say this is a feature by Professor Whitesides from Harvard University. I'm happy to say that I was invited this and published this year a feature in analytical chemistry in which I report the several advantages on using the paper-based electrochemical device instead of the classical one. I would like to thank all funding, all the people that work with me, Luca, Vincenzo, Laura, Nargis, Afid, Chef, Ludovica, Christian, Laura, Giorgia, Rachele, and the people from Sense4Med, Arianna, Matteo, Francesco, Veronica, and Noemi, and all of you for the kind attention.
Thank you very much, Fabiana. It was a very nice presentation with a lot of information. And because we have just a little time, I want to ask if there are questions from room. Please, go ahead.
Thank you very much, Fabiana. Very interesting presentation. I was interested in the section with the biosensor for identification of botulinum toxin. You mentioned that you identify with the smartphone assisted analysis. Exactly. How do you do this part? Did you download any program, and how you connect the paper, the biosensor to the smartphone? How you receive the information from to the smartphone?
Yeah, thank you for the question. So this potentiostat miniaturized one is commercially available by PalmSense instrument. So is connected with the smartphone, in which we have the app. For example, we are working to have a system for cystic fibrosis. Also, PalmSense is able to customize the software, the app. But now, if you buy this device, we have also the possibility to download the app by PalmSense website. And we connect easily by 3-pin this paper device inside the Sensit portable potentiostat miniaturized one. You can say that also this device can be also produced by really for mass production and also customized by the PalmSense in function of the application.
So you want to say that the device is the main key to identify?
This device is a miniaturized potentiostat. So you can use this device in any electrochemical technique. In my case, for example, I detect botulinum toxin with the electrochemical probe using square wave. So using the app, we can select the square wave, make the measurement. If you customize another sensor for another application, for example, we also develop for pesticide detection in amperometric mode. Also, you can use the same system, so smartphone assisted. By the app, you can select the amperometric mode, apply the condition, make the measurement. So it's a very nice product. So help us to really produce a miniaturized device, but made, of course, by electronic engineering by PalmSense, and is commercially available. You can buy by PalmSense.
Thank you, Fabiana. Thank you.
You're welcome.
Thank you very much. If there are other questions, maybe from online, if there are questions.
Any questions?
I have a lot of questions, Fabiana, but I will resume just to one, a general one. From your experience, the time from the idea to market for a sensor for medical application, let's say, is around how many years?
Yeah. So for the just because it's a very thanks for the comment. It's a very important comment because sometimes also in several conferences we say to reach the market, the most difficult things is the mass production. But I can say no. The really in biomedical field, the really problem is the regulatory aspect. So means the clinical trial, especially because now in Europe, the last year, it changed the regulatory aspect. So it's the regulatory is now is very not so easy. Reason for that, some also European companies start for the American approval because now is easier. And so because we have to respect all the safety condition, and in addition, a huge budget is for clinical trial. So means that if from academic point of view, the most difficult things before, we can say the mass production, but we can say no. The most difficult things is start with a huge budget, the clinical trial, to respect also the regulatory aspect. The reason for that now, not so easy, because really, if you check the literature, you have a lot of point-of-care device, but in the market very few. Now glucose strip, for the COVID, the pregnant test, not so much. And so the reason what I think also when, for example, discuss with other colleagues that work in this TR level is that part. So I would like to boost also other colleagues to take this direction, because if you're able to deliver point-of-care device to the society, really we are able to make something that can help the people. Reason for that, we select rare disease, because for the cystic fibrosis diagnosis, the sweat analysis, so chloride content in sweat is a reference analysis to start the clinical trial for make the drugs, also for the baby. And you can say that not so many laboratory make this test. For example, in Italy, some people from Sicily have to fly every month, for example, to go in Rome for Bambino Gesù Hospital to make the analysis. So we have the possibility really to improve the life of some patients. What we did for glucose strip.
Thank you very much for your comment, Fabiana. And if there are no other questions, we will end here the first plenary session, and we'll have 10 minutes or 15 minutes of break, a coffee break. Thank you again, Fabiana.
Thank you. Thank you for the invitation, Michaela. Thank you so much. Bye.
So welcome from the short break. I hope also our colleagues from around the room will hear us. So right now we will start our regular presentations with the oral session. They are a mix of the two sections. You will see all over the program. We didn't maintain the presentations by section, but more in order to keep your attention, it's a mix of the two sections. And I will pass the microphone to my colleagues, Fabiana Arduini and Andrei Sârbu, to chair this section, please.
Thank you. So we will first start with the presentation of Professor Barkat Ali Khan. I hope he is connected. Please respond if you are there. Okay, so probably he will connect later, or we will do something. We will contact him to present his work. So we probably will go to the next presentation. The next presenter is here physically, Alexandra Luzina. The title of her presentation is FAPA-MS, Enhanced by Molecularly Imprinted Polymers for Targeted Environmental Analysis.
Okay, thank you very much for introducing me, and thank you for the opportunity of presenting my work here today. So as was mentioned, my name is Alexandra Luzina, and I'm a PhD student from Adam Mickiewicz University in Poznań, Poland. And also I am a member of a supramolecular chemistry group there. So today during my presentation, I will outline the synthesis, characterization, and also the application of poly-2-oxazoline-based molecularly imprinted polymers called MIPS for targeted environmental analysis. So basically, the general aim of my whole PhD research is, at first, the synthesis of a functionalized materials, functionalized poly-2-oxazoline-based MIPS, and then, secondly, use those materials in a rapid and selective detection on selected analytes, especially those analytes which are hazardous to the aquatic environment and in the combination of the FAPA-MS technique. But why poly-2-oxazolines? Because of the beneficial factors like biocompatibility, like stealth behavior, but from my point of view, the high functionalization possibilities. And on the bottom of this slide, I'm showing you the couple of the examples of 2-oxazoline derivatives, which are currently the subject of a supramolecular chemistry group of research. And I marked 2 of them in red because these 2-oxazolines are currently the subject of my PhD research. And in the next part of the presentation, I will focusing on these 2 derivatives. And why MIPS? Because in comparison to other well-recognition systems, MIPS have gained a huge scientific interest. And the widespread use of MIPS is the aftermath of the favorable characteristics like high stability in aqueous chemical and physical conditions, like a remarkable robustness, relatively low-cost synthesis, and straightforward synthesis. And what MIPS are? These are the special polymeric systems that possess the crucial ability of the selective recognition of a desired templates. This crucial ability is determined during the MIPS synthesis because during the synthesis, the polymer matrix is being mixed together with a template molecules. And that goes in a creation of a special MIPS interactions between these 2 species. Basically, these are the non-covalent interactions, and the hydrogen bond is the most common. So after obtaining this whole system, the template molecule can be easily removed with the use of dialysis, for an example, and that goes finally in a creation of a special MIPS cavities, which should be selective toward our specified templates and should be able to rebind these templates for, as example, matrix environmental samples. And on the slide, I'm showing you the key steps of my PhD studies. And I will try to make a quick explanation of each of the steps. At first, template selection. The template needs to correspond for both, for the polymer matrix, but also the functionalities that comes from the functional agents and cross-linking agents. And given that our research group is focusing on a water treatment, we need to find some of the examples of the compounds that may be easily found within the wastewater. And unfortunately, the list is quite long. However, I'm showing you some of the classes of compounds and the exact compounds, which are currently the subject of a supramolecular chemistry group of research, and especially those kind of research in which I'm involved in. Then polymer synthesis. So I'm focusing on a butanox and C2-mestox, as I've mentioned previously. And the goal for me is to obtain the polymer with the terminated double bond at the side chain. As you can see here, butanox possesses it. So after the polymerization, I can just directly use it for the modification. Unfortunately, C2-mestox do not possess it. So it means that I need to make modifications. So after the polymerization part, I've made a side chain modification with the use of a allyl amine and obtaining the polyallyl amidox with a terminated double bond on the side chain. And this part of my work was performed in the collaboration during the internship at Ghent University. Then modification. So functionalization part with the use of a thiol and click reaction with the UV radiation exposure, with the use of thiols, here as an example, trimercaptopropionic acid. The materials were differentiated by the degree of functionalization. And as you can see here, I'm showing you the proton NMR spectra after this process. And there is an extra signal around 1.5, which were used for the calculation of a real functionalization degrees. Then cross-linking part with the use of the same methodology, but with the use of a dithiols and with the use of template or without template for shaping MIPS and NIPS respectively, and with the use of the remaining double bonds after the modification part. So here is the example of a matrix compound, so butanox with the MCPA molecule inside, And after obtaining this whole system, we can just make the template removal with the use of a dialysis, so shaping the remembering cavity. Then the adsorption study is the final part of my work. And I'm showing you here the total list of all of the adsorption studies, which I used to do. And I want to make a stop within the FAPA-MS technique. And the FAPA means the flowing atmospheric pressure afterglow combined with the mass spectrometer. So plasma-based MS involves introducing sample into the plasma stream, which ionize the sample and then transfer the ionized species into the MS inlet. So it means that I am able to obtain exact mass spectrum in a second and without any sample retreatment. So in my studies, I'm placing MIPS and non-imprinting materials called NIPS in an electric heater beneath the FAPA plasma ion stream. And when the temperature increasing, the sample underwent the thermal desorption and ionization upon reaching the plasma ion stream. So here are the results from the FAPA-MS, limits of detection, which is quite low, as you can see, especially in comparison to the direct solution analysis. And the linearity, which is also quite broad, because it's from LOD values to the value over 100, beyond which oversaturation occurred. And the general conclusions are that for the MCPA MIP 30 degree of functionalization with a 3-mercaptopropanic acid, a 200-fold improvement of LOD value was achieved. Then the quantification of a proper paraben enhanced sensitivity by 2 orders of a magnitude. Sensitive detection of explosives gives us the LOD down to 0.01 micromoles, and the PFOS, so the fluorinated surfactant, was detected at levels as low as 5 ppb. And from the other results, so let me follow the MCPA example of article. We can make the adsorption isotherm tests. So we can then fit the data with some of the mathematical models and then also calculate the maximum adsorption capacity, which is quite important when we're thinking about the switching into the industrial application. So the MCPA here follows the Langmuir model. And also I want to mention here that the material with the most effective adsorption value is the material of a 30 degree of functionalization, so the green light. And also when we're switching into the selectivity, it's also the most effective material. And the selectivity was performed in a competitive measurements with the use of the similarly compounds like herbicide 2,4-D. So in summary, the short-chain 2-oxazoline-based polymers are well suited for the synthesis of a functional materials used in a MIPS fabrication. In general, the use of carboxylic acid groups and also increasing the functionalization part shaping the more effective adsorption parameter. Then polyoxazoline-based MIPS enables sensitive analyte detection with the use of a FAPA-MS technique, and the use of a MIPS allows for a substantial reduction in an LOD's value, especially in comparison to the direct solution analysis. The innovative FAPA-MS techniques enables selective detection within seconds and without any sample retreatment. So the perspectives are the use of alternative classes of compounds for the templates molecule, like pharmaceuticals, preservatives, or other things like PFOS, and this article is under preparation now. Use some unconventional crosslinkers like boron compounds, or also make a combination between porous materials and also the MIPS for shaping a more effective adsorption parameter. And one of the most promising perspectives is the use of these materials for a sustained thermally controlled release of some of the active compounds for plants. And this part marked in green here was performed here at ICECHIM during my first stay here in spring. So now I'm focusing on a sustained release with the use of the same compounds. Thank you very much for your attention.
Thank you very much for the presentation. And now we can take some questions from the participants, if you have any question.
I have one question. If I well understood, you perform this process like this. You have the polymerization, then functionalization. And when do you put to template?
The template is being putting within the cross-linking part, because in comparison to other well recognition MIPs like a metacrylate, it can be done directly from the monomer to the MIP.
Yes.
And we cannot do that with the poly-2-oxazolines because
Exactly.
poly-2-oxazolines are very weak with the nucleophiles. So we need to prepare at first prepolymer, make the modification, and then make a cross-linking to MIP. And within this part, I'm placing there the template molecule.
Yes, I understood. It's a good idea. Thank you.
Thank you.
Thank you. Other questions? Or is there anyone online that wants to ask any questions?
Good afternoon. Very interesting research. Thank you. No question.
Thank you. Okay. Thank you very much. We are going to ask again if Professor Khan has connected. Okay. So he's not connected yet.
So we will pass to the next presentation. It's a friend of ICECHIM, which will perform this presentation, Lucia Pintilie. And the team is Lucia Pintilie, Maria Mangan, Constantin Drăghici, Miron Teodor Căpruiu, Mădălina Diana Gaboreanu, Ioana Cristina Marinaș. The title is The Design and Development of Some Fluoroquinolone with Activity Against Multi-Drug Resistant Microorganisms. Please, Lucica.
Yes,
please,
thank
next
you.
presentation.
First of all, I want to thank you for the opportunity to be again here. Yeah. The scope of this work was because the resistance developed by bacteria threatened virtually all classes of antibacterial agents. The economic impact of antibiotic resistance is considerable and will continue to grow. European Union estimates that antibiotic resistance causes 600 million days of lost productivity each year. A post-antibiotic era means the end of modern medicine, as we know. Things as common as strep throat or a child's strep knee could kill again. In this moment, identifying a new molecule with activity against multi-drug resistant microorganisms and developing it into a new drug would be an important step in the fight against antibiotic resistance, leading to improved health and life expectancy of the population, reduce medical costs, and increase the productivity. This work presents the preparation of new compound with activity against multi-drug resistant microorganisms, molecular design studies and molecular docking studies, also present chemical synthesis study for the compound selected to have a predicted good biological activity, confirmed the structure of the new compounds, and evaluated the biological activity. The compound who has proposed to be studies are presented in figure 1. It's a quinolone compound who has in 7th position the substituent presented in this table. First step was the molecular design study. The first step of this study was the preparation of the ligand, who have been prepared with using Spartan 24 software package. We have determined we optimized the geometry of each ligand, and we have calculated a sum of electronic properties. The optimized structure of the 6 compounds are presented in this figure, and the molecular property are presented in this table. The energy of HOMO and LUMO orbitals, the PCA, ovality, hydrogen bond acceptor, hydrogen bond donor. In this slide are presented one of these compound, optimized molecular structure of a compound FPQ102. And here are presented some surface who help us to visualize the mode of interaction of the ligand with the protein receptor. Here are presented the HOMO and LUMO surface and the difference between energy of HOMO and energy of LUMO, who show us the compound have a high stability and low chemical reactivity. Molecular docking study have been realized with 3 software: CLC Drug Discovery Workbench, Molegro Virtual Docker software, and Gold software. And the interaction, study of design compound has been performed with the protein receptor Staphylococcus aureus DNA girase subunity A, who was downloaded from Protein Data Bank. The results obtained with the software are presented. All 3 software, here are CLC Drug Discovery Workbench. We have determined the group of interaction, the hydrogen bond. We measure the bond length for each ligand. And with this software, it was observed most of compound realized the hydrogen bond with the amino acid of interest, serine 84, who is the same amino acid who realized the hydrogen bond, moxifloxacin, who is the ligand of interest. Also, with the Molegro Virtual Docker software, we obtained some results. Here we can visualize also the electrostatic interaction and the steric interactions of each ligand. Also, we have determined the hydrogen bond and the electrostatic or steric interaction realized by each ligand. With the Gold software, some study has performed, and after analyze the result obtained, we obtained some results. We obtained results or who are put it in this diagram. The compound have been synthesized by Gould-Jacobs method, who consist in reaction of condensation and cyclization of anilines, trifluoro-4-chloroanilin with etoxymethylene malonate, and introducing the substituent in first position. And we have realized the hydrolysis of the ester group for obtain the K intermediate, the quinolone acid, 3-carboxylic acid. And after that, we have some amination reaction for introducing the substituent in 7 position. The compound obtained have been characterized by NMR spectra, TIR spectra, and also another analysis. After that, we have realized the evaluation of antimicrobial activity. First of all, has been evaluated the qualitative evaluation. The most active compound against gram-positive and gram-negative bacteria were found the FPQ 202 and the FPQ 203, and was observed that only FPQ 201, 205, 206 showed antifungal activity against the Candida albicans. We evaluate the quantitative antimicrobial activity. The results are shown in this table. We was observed most active compound, the all compound have been active on gram-positive and gram-positive bacteria and gram-negative bacteria. And it was observed gram-positive bacteria was active only Pseudomonas no, sorry. K. pneumoniae, Klebsiella pneumoniae, and the Pseudomonas aeruginosa. The best effect was observed for FPQ 202 and the FPQ 203 compound. Antimicrobial activity on Candida albicans is similar to that of the solvent used, dimethyl sulfoxide. They don't have activity against Candida albicans. It is known the quinolone don't have activity against fungus. Here are presented some, and in this figure are presented the results of evaluation of antimicrobial activity. Also have been studied the influence on microbial adhesion, which represent the initial stage of microbial biofilm development. The value of the inhibitory concentration for microbial adhesion MBAC are presented in table 4, and the compound who has the MBC value lower than MIC are considered active for inhibiting microbial adhesion. Most of the compound inhibit the adhesion of Enterococcus faecalis cells. In conclusion, we have designed some quinolone derivatives. We realized drug design and molecular docking simulation. We obtained by synthesis dense compound. We confirmed their structures, and we observed a correlation between the predicted data obtained by molecular docking study and the experimental data obtained from the evaluation of the antimicrobial activity. Oh, sorry, no antitumor activity, sorry. And I finished my presentation. Thank you very much.
Thank you for your presentation, very interesting. If there are questions, if not, I have one. Do you know something about the mechanism by which these drugs are killing the yeast or bacteria?
Yes, they inhibit the adenogirasa and topoisomerasa 4.
Okay.
This is their mechanism.
And how they are entering into the cell?
They created
are able to pass the membrane?
Yes, yes.
Because they are not too big?
Yes. And they created some hydrogen bond with the amino acid, and realized the decatenation reaction, rupere de amino acid. Yes, and they kill bacteria.
Okay.
This is the mechanism. But they also have installed antimicrobial resistance.
Yes, I understood. Is there other questions from online? If not, we pass to the next presentation. Thank you.
Okay, so the next presentation is of Rakim Zan Kosanov with a presentation, computational study of effectiveness of third amyl methyl ether and ethynyl cyclopentanol as octane boosting additive in gasoline. Okay, so you're online. Thank you.
Good afternoon. dear researchers, conference participants. I am Kosan Brahmjan. I am from Kazakhstan, and I am a teacher in a school. Yeah, I am studying at university for master, and I am teaching in school also. So I present at our university and oil and gas together with my researcher is a professor and Dr. Amajan Sagnaev, and my third professor is Amangelde Ibrahim. She is also assistant professor. So my research topic is increasing octane number of these petrol, gasoline, yeah, benzene, gasoline. So I am researching now is different like compounds. Today I want to compare these two compounds, how they are increasing octane number of petroleum, yeah, gasoline. It's just my like theoretical researcher. I am like now is going do a practical part. Yeah, I'm like order some equipments reagents for my practice part. First, I want to express my thank you for Ministry of Education. Yeah. Ministry of Education and Researchers, National Director of Research organizing Romania. Thank you for your like organizing like this very good symposium. Yeah. And thank you for partners, yeah, publishing partners, and for sponsors also. So because like the conference very important for us like researchers. Okay, about like gasoline, yeah, you know everyone now is the gasoline. Very famous, yeah, very famous mixture, yeah, liquid mixture of light. But is now is in more countries they are want to need a new part of oil, but you know in our country it's famous. We have like a lot of cars. I don't know, maybe after 10 years, 15 years, we will like need in our country like another type of cars, like either with H2 or like electricity. Yeah, but now is important like our air also. For that, we are like researching this topic. So what about octane number? Octane number indicates the resistance of gasoline to knocking during combustion, because it's not less number in their combustion will be like when they combust, they will detonate the engine, yeah, car engine. So I comparison this. I researched some like in the world their octane ratings, yeah, in different countries like Australia, Japan, Europe, USA also. So in USA, octane number is I found this research in one research, yeah, report. So I don't know. It's maybe it's correct, it's incorrect, but I think, in my opinion, it's not like good, because look, 87, I think is less than others, because in my country, minimum 92, 95 we are using, like Europe, Japan, Australia, and more than 90. Why not more than 90? So okay. What about my research aim? Yeah. Is it TAME is a tertiary amyl methyl ether, and the second one is my compound, the ethylene cyclopentanol. It's one ethylene cyclopentanol. Yeah, before some researchers they are already using TAME, tertiary amyl methyl ether, for their gasoline, but I want to check. Yeah, I am researching ethylene cyclopentanol, how is affecting for these gasoline octane numbers. So because you know, the environmental process is very like important, and Here you can see their like emission, yeah. If their factor number is good for air, for environment, will be like get less less gases, yeah, like carbon dioxide and SO2 or NOx, yeah, NOx. So it depends for combustion, their combustion also. So in this table, I compare these two compounds. Yeah, first their structural formula. My compound, the ethinyl cyclopentanol. These are like cycle compound. So cycle compound, it means their combustion very like not faster. Yeah, for faster, it means we can like save petrol. Yeah, gasoline. It's also important. Second one is in my compound we have a like triple bonds. Yeah, triple bonds means it will be like stable than either. For that, in my opinion, ethynyl cyclopentanol will be help for increasing octane number of gasoline. Yeah, gasoline. So water solubility is there both also lower, yeah, because they are look in a TAME is a more part of this compounds like non-polarity. For that, it's solubility of in water very low, like my ethynyl cyclopentanol compound also. So what I researched a lot of like researcher, yeah, their structure, their formula, boiling point, molecular weight, yeah, their polarity also it's also important because they will write in a car engine with oxygen, yeah, in oxygen. I calculated with my math teacher, yeah, with my teacher there. It's like theoretical, yeah. I will check when I will do their. I will add in the future, I will mate first and I will add in a gasoline. After that, I will check again these dates. Yeah. Now is if I compare with Tănase, is my ethynyl cyclopentanol, it's good. Yeah. Because effectiveness also good, stability also high, bond energy also higher than Tănase. Yeah. So it will be like it will be helpful for my gasoline. So gasoline. So about oxygenates, yes, they are very famous. They are very famous. So I researched again another like oxygenates like methyl tert butyl ether, yeah, ethanol also. They are stable because they are aliphatic. They are available. We can make ethanol, for example, with like corn with from sugar, like something, yeah. So what about ethynyl cyclopentanol? It's like higher octane boosting, yeah, in my opinion, because it's they are structuring like showing like this, and the lower volatile it's also like important, and their potential more environmentally friendly, and but not widespread in industry. Maybe in the after me, in maybe some countries also they are researching. Yeah, I hope because it's a very interesting compound. So how I will get this compound in my laboratory? Yeah, so I am preparing now with the practical part. Yeah.
Sorry, you have 3 more minutes, so maybe you can go quicker.
Okay. So it's my plan. Yeah, I have a catalyst. I have a solvent ethanol. I ordered my like reagents. Yeah, ethylene gas and anything. So cyclopentanol I ordered. So but I will do. Yeah, it's my like reaction. So it's also like they are infrared. How I will now is I get my compound. Yeah, I will check there with infrared. this carbon NMR also, proton NMR also. Yeah. So in conclusion, theoretical part of giving for me, yeah, my compounds, it's an cyclopentadol, it will be like very helpful, yeah, increasing octal number more than TAME, yeah, more than TAME. For example, if I will add the TAME for gasoline, it will be like improving around 6.2 units, yeah. If I will the ethylene cyclopentadol, it's will be higher than TAME. It's around 8-9 points, yeah, increasing octal number. It means it will be like combustion in car engine, very like completely combustion. It means it's a low toxicity and a high environmental friendliness in an environment also. So and I can use my compound. I can like help for ecological also. So thank you. It's my references when I use it for my research. Thank you.
Thank you.
Questions?
Thank you for your presentation. If you have any questions for our participant or our questions from the online, I want to ask you something. Have you tried using both of them together?
Sorry, my professors here, yesterday, 2 days before, they went to another conference, yeah, because before we don't know when we will like our this symposium. For that, we like divided for 2 conferences. Sorry.
Okay, thank you.
Hello,
Thank
we
you.
now pass to the next presentation. It's Miruna Ioana Sava, Michaela Deaconu, Cristian Matei, Daniela Berger. The name is Ulva lactuca, a source of polysaccharides for gold nanoparticle synthesis. Please. You have 10 minutes, yes, or less, if possible.
I have laryngitis, so it's an audio on the presentation.
Doriți profe, nu le cercăm banane. Guardo una se fare profe, volete le avanzare?
Ah.
Perché?
Che succede che la stagione viene avanzata
Vă rugăm să închideți microfoanele, cei care sunteți online. Close the microphones. Close the microphone for online.
Thank you. It is a lot of work. If there are some questions, I have one question. Why do you say that you have nanoparticles? I saw that the mean size is 500 nanometers. These are almost.
They were 47 and 71 nanometers.
Okay.
Yes.
Yes. You are right. Thank you. Yes, I saw now the lab.
Yes.
Right. If there are other questions, if not, congratulations and good luck.
So for the next presentation, we have Alin Gabriel Gheorghe with the title Properties of Biocomposite Materials Containing Silver and Gold Nanoparticles.
Thank you very much for introducing me. Today I will talk about mesoporous silica modified with silver and gold nanoparticles to use as carrier for phytocompounds from a wild bilberry leaves extract. Mesoporous silica has been chosen as support for its outstanding properties, such as high pore volume, tunable morphology, the versatility of its surface properties, and good biocompatibility. Here you can see that the surface of the mesoporous silica can be functionalized with various biomolecules, such as antibodies and proteins, as well as metallic nanoparticles, which widens the range of possible applications. Lately, gold nanoparticles have been used in diagnosis and treatment methods such as point-of-care testing and photothermal therapy. And they also have good biocompatibility, and the surface of the nanoparticles can be loaded with various bioactive compounds. Silver nanoparticles have antimicrobial properties against a wide range of pathogens. They also are used in targeted drug delivery, and the disadvantage is the cytotoxic effect on cellular level, depending on the dose. The aim of the work was the synthesis of functionalized mesoporous silica modified with gold or silver nanoparticles for further using as carrier of polyphenols from an extract obtained from wild bilberry leaves. We used two types of supports. The first one, MCM41, was synthesized using the hydrolysis of tetraethyl orthosilicate in the presence of base catalyst triethanolamine and surface directing agent acetyl trimethyl ammonium chloride, which was later removed by calcination. Then the silica was functionalized using aminopropyl triethoxysilane by the post-synthesis approach. The second support, pitaya type silica with larger pores and thiol moieties, was obtained by the co-condensation method. The basic medium was ensured by sodium salicylate, and the surface directing agent was acetyl trimethyl ammonium bromide, which was removed later by extraction in acidic ethanol. Silver nanoparticles were obtained by reducing silver nitrate with sodium borohydride, and gold nanoparticles by reducing tetrachloroauric acid with sodium citrate. Here you can see the position of plasma resonance absorption maxima for the resulting colloids. Then the amine functionalized silica was impregnated with the silver nanoparticles, and the thiol silica was modified with gold nanoparticles. The wild bilberry leaves extract was prepared using a solvothermal treatment in ethanol at 60 degrees Celsius and 5 atmospheres of argon pressure. Then the extract was encapsulated in the mesopores of the modified silica. Scanning electron microscopy images of the functionalized silica reveal uniform morphology and particle sizes of about 100 nanometers for the MCM type support and 130 nanometers for the pitaya type silica. Transmission electron microscopy images of the silica modified with the metallic nanoparticles show that the size of the silver nanoparticles are about 5 to 7 nanometers, and the gold nanoparticles are about 20 nanometers, and they are well dispersed in the pores of this support. X-ray diffraction analysis proved the existence of the metallic nanoparticles on the surface of the silica. The wild bilberry leaves extract was characterized using spectrophotometric determinations such as total polyphenolic, flavonoid, and tannin contents, as well as radical scavenger activity by two methods, DPPH and ABTS. By high-performance liquid chromatography from 23 standard compounds, only 4 were quantified. Catechin hydrate, which has anti-diabetic potential, chlorogenic acid and rutin hydrate with well-known antimicrobial properties, and cumaric acid. In order to determine the concentration of the extract in the biocomposites, we used thermogravimetric analysis, and the silver-based carrier had more extract encapsulated than the gold one. Biological evaluation, biocompatibility and antioxidant activity on cellular level were assessed on human keratinocytes, and a good cell viability was reported even after 24 hours and the higher concentration of the biocomposite. And a better reduction of reactive oxygen species levels when the extract was encapsulated in the proposed carriers. Anti-inflammatory activity was evaluated on THP1 cells, which were previously inflamed with lipopolysaccharides. And the samples containing gold secreted a lower amount of pro-inflammatory cytokines, TNF-alpha. Regarding the antimicrobial properties, both of the supports were effective against methicillin-resistant Staphylococcus aureus, and only the silver one against E. coli. Conclusions: developed mesoporous silica modified with silver or gold nanoparticles can be employed as carriers for various phyto compounds. The developed biocomposites containing wild bilberry leaves extract exhibited good antioxidant and anti-inflammatory properties and perspectives. Based on the biological properties of the developed biocomposites, they can be further included in topical formulations.
Thank you very much for the presentation.
Thank you.
So if we have any questions from the participants or online? No, so everybody understood. So maybe I have a very short question. So you used only 2 types of sealants for preparing the nanocomposites. So I was wondering what is the difference between the 2 sealants, or why 2 different directions? So why didn't you use other type of sealant monomer for your
functionalization?
Yeah.
Because those were the ones that we had.
Okay. Yes, we know this answer. Okay.
Yes, these were the ones.
And I believe the one based on mercapto moiety, you used it for silver particles.
The
basically.
thiol one was used for the gold particles and the amine one for the silver.
Ah, okay. Okay, thank you very much.
Thank you.
Okay, now we performed the last presentation. It's Livia Teodora Ciobanu, Diana Constantinescu Aruxandei, Florin Oancea, Ileana Cornelia Fărcășanu. The title is East Surface Display of Fungal Cysteine-Rich Proteins for Biogenic Selenium Nanoparticles Production.
Thank you. Okay, so for the surface display of fungal cysteine-rich proteins in yeast for production of biogenic selenium nanoparticles. Our research aims to provide solutions for the paradoxical issue of selenium, which is both essential and toxic. It is essential in humans and animals because it is incorporated in the form of cyanocysteine in cyanoproteins. These proteins have a crucial role in maintaining the oxidoredox balance. They are involved in various processes of peroxide reduction. They regulate the thyroid hormones, provide neural protection, preserve reproductive health and the good functioning of the immune system, and so on. In plants, however, selenium has not been yet classified as an essential nutrient, but it seems that plants need it since they accumulated in the form of cyanomethionine. This compound has been associated with the improvement of the resistance of plants to abiotic stress, to biotic stress, also since it is involved in pathogen defense. Selenium is a growth regulator in plants and also an antioxidant agent capable of scavenging reactive oxygen species. However, high dosages of selenium can trigger pro-oxidant effects and can even lead to acute toxicity. These high dosages are pretty close to the recommended ones, and here we've come to the very main issue of this element, which is its narrow safety margin. This issue makes it hard to handle the selenium deficiencies. This is an understated problem. At the moment, globally, it affects mainly China and Eastern Europe countries, including Romania. It is due to poor dietary intake of selenium. Basically, the soils are deficient in selenium, and so are implicitly the crops. A solution would be to employ selenium biofortification in favor of preserving the good health of humans, animals, and the good functioning of various plant ecosystems. Okay, it seems like it's not working. Maybe. Sorry. Okay, I'm gonna use the laptop. Fortunately, advances on this research field have established that selenium nanoparticles are the least toxic form of selenium. They are composed of zero-valent selenium, which is the state of this element that exhibits the lowest toxicity. Moreover, they provide the advantage of a higher reactive surface. Throughout the available synthesis methods of nanoparticles, chemical, physical, biological, the biological ones clearly stand out. They are more environmentally friendly. They refer to the ability of microorganisms to reduce selenium species to zero-valent nanoselenium. Moreover, these synthesis hosts are able to provide biomolecules which can cape the nanoparticles, stabilize them, and provide certain bioactive properties derived from the system wherein formed. Therefore, in this context, our experimental approach aims to use yeasts as for the production of selenium nanoparticles, but we have genetically modified them, trying to transform them into sort of nanofactories for the production of customized selenium nanoparticles with enhanced stability and enhanced biological activity. We have done this by cloning of cysteine-rich sequences into yeast, specifically Saccharomyces cerevisiae yeast strain. We have focused on hydrophobins and ceratoplatanins. These are proteins specific to fungi. The corresponding genes have been isolated from Trichoderma raceyi strain. They have special properties. They are surface active. They have the capacity to self-assemble at on surfaces and modify their interfacial tension. Moreover, hydrophobins have already been studied as effective caping agents for nanoparticles. To the best of our knowledge, ceratoplatanins have been not, but they share some properties with hydrophobins, which we think that this would make them efficient caping agents for our selenium nanoparticles. Also, this kind of synthesis could expand the applications of these nanostructures, since selenium nanoparticles have been already identified as plant biostimulants, but also hydrophobins and ceratoplatanins can act as elicitors in plants, triggering their immune system, and therefore they could become novel and interesting alternatives as plant biostimulants. Before presenting the actual results, I'm going to state out that we have abbreviated each protein because they are pretty new. They have only the unique identifier number, so we provided them an abbreviation. In the case of hydrophobin 1, it is already a standard abbreviation HPB1. We have also hydrophobin, which is a hypothetical protein. We have abbreviated it HYPE, and the ceratoplatanins are abbreviated E1, E2, C65, and C82. Our aim was not only to clone and to express these proteins in yeast, but to display them at the surface of yeast cells. We have therefore worked with specifically designed plasmid for yeast surface display, namely PID1. We have begun by designing primers to amplify the target genes. We have designed 2 sets of primers for each target genes, one which would allow the translation of the insert within the full length of the plasmid. So they have the start and stop codons, and this would provide the versatility of cloning in any plasmid, but we have focused on PID1 for now. The second set of primers is sort of specifically designed for the cloning in PID1 and for the translation of the insert within the incomplete coding sequence of the plasmid without certain downstream vector encoded elements. We have integrated also restriction sites for 2 enzymes, BMH1 and EcoR1. This will allow the enzymatic cleavage during the cloning process, and it will allow the integration of our gene in the plasmid sequence, somewhere around here in this red square where it is pointed with an arrow. I don't know if it can be seen very Very good. It is right next to an enterokinase cleavage site. These plasmids provide various sites for cleavage of or identification of our proteins. This allows separation and further characterization of the expressed protein. The first step of the cloning process was to amplify and purify the target genes. We have primarily begun by extracting RNA from Trichoderma racea mycelium, which was obtained by growing the strain in an inductive selective media, which allowed the expression of ceratoplatanins and hydrophobins. We have used this RNA as a template for complementary DNA synthesis, and this cDNA along with the synthesized primers to amplify the target genes through reverse transcription polymerase chain reaction. The products were purified using a DNA purification kit, and we have loaded them onto an electrophoresis agarose gel. The obtained bands were of expected size, 0.5 kilobases for the most part, with an exception in the case of a ceratoplatanine C65, which is 0.7, 0.8 approximately. The next step was the cleavage of the target genes and in parallel the cleavage of the plasmid. We performed this step in order to provide cohesive ends for each sequences, which will allow the joining together of these two products. We loaded the digestion products on an electrophoresis gel, and considering the band morphology, especially the slightly smeared edges, we have concluded that the cleavage has been achieved, and so we proceeded to the ligation step, which is the joining together of the cleaved target gene and the cleaved plasmid, which now have cohesive ends and can stick. We have obtained, in the case of each sample, a single band, an intense band for each sample, which is also slightly higher than the empty plasmid, which confirms that indeed the plasmid contains an insert. However, the difference is not substantial, of course, because the inserts are very small and the plasmid is 5.0 kilobases. But the presence itself of a single band, it's enough to confirm that we have obtained recombinant DNA. We use each ligation mix to transform E. coli cells for further isolation and screening of recombinant DNA. Each E. coli transformant was plated on an agarized Luria-Bertani media supplemented with ampicillin, which served as a selective marker because PID1 plasmid has an ampicillin resistance gene, therefore allowing this kind of selection. We have selected 12 to 24 colonies from each plate. We have cultured them in fresh LB media, and we have isolated recombinant DNA through alkaline lysis method. It's that in the case of the constructs designed for the incomplete translation, the translation of incomplete sequences in PID1, it was a very good yield, a very good cloning yield. So we have selected from each group a positive sample for further purification. It is important to mention here that we chose to digest the recombinant DNA with the same enzymes which we used in the first step of the cloning process in order to allow the release of the insert and to visualize it along with the plasmid. So all of these are our inserts. This is the plasmid. We have selected from each group a positive sample, only one, because that's all we need for further purification of recombinant DNA. The next constructs, those with which allow the translation of the gene within the full sequence of the plasmid, had a rather lower cloning yield, but we still managed to select positive sample from each group. And we have then selected the E. coli colonies, which correspond to the positive samples. We have cultured them again, and we extracted the recombinant DNA once again, but this time using a purification kit for extraction of plasmidial DNA. We have once again digested the recombinant DNA and loaded it on an electrophoresis, and we have confirmed the presence of the plasmid and the insert in each sample. So we have proceeded to use this recombinant DNA for the transformation of Saccharomyces cerevisiae yeast strain. These are the transformants obtained from each chimeric construct, and we have obtained them by cultivating the transformants on minimal glucose media containing yeast nitrogen base, cas amino acids, and glucose, and then we have selected from each plate a colony and propagated it on the same minimal media to generate stock cultures. We have made a preliminary test to assess the effect of sodium selenite, which is a selenium nanoparticle precursor, to assess the effect of sodium selenite on the growth of Saccharomyces cerevisiae transformants. We have cultured each of these strains, each of these constructs on minimal glucose media, and then transferred them into media with galactose to trigger the gene expression under the GAL1 promoter from the PIL1 plasmid. And we have serially diluted each culture and then stamped it on galactose media. The preliminary results showed us that there is a certain strain transformed with an E2 ceratoplatinum, which has the ability to reduce sodium selenite to sodium nanoparticles way faster than other strains. This is confirmed by the red coloration, which indicates that the sodium has been reduced and sodium nanoparticles have formed. This experiment also offers us some information about the sensitivity or the increased tolerance of certain strains. For example, this strain seems to be among the most sensitive to sodium selenite along with hydrophobins, while this one, which was also able to reduce sodium selenite at high concentration, was also the most resistant one. And it's interesting that some morphological differences of the culture are observed. But of course, we will proceed to make further tests to assess these results. Thank you so much for your attention. And if there are any questions or suggestions.
Thank you for your presentation. If there are questions from online. If not, congratulations.
Thank you very much.
And we stop here. We have a break till 14 hours, and we will be back at 2:00 p.m.
And for the on-site participants, please, you are all invited to the lunch. It is on the third floor, is the lunch room, so it will be a lunch between colleagues and friends. Thank you. Welcome back from the lunch break, everyone. I hope everyone had a very productive discussion with their colleagues and their partners. We will start the afternoon session with two very, very interesting invited lectures. First of all, it is, I hope Alvina is online. It is a very big honor for me to present the next speaker. Alvina Avagyan is from the Scientific Center of Vegetable and Industrial Crops, Darakert, Ararat Mars in Armenia. Dr. Alvina Avagyan has nearly 30 years of experience in the conservation and the sustainable use of crop diversity. She currently serves as a lead researcher and gene bank manager at the Scientific Center of Vegetable and Industrial Crops. Alvina is a breeder and co-author of several varieties of emmer wheat and vegetable crops and has published over 110 research papers in this area. She holds a PhD in biology and graduated from the Faculty of Biology of Yerevan State University and Armenian Agricultural Institute. In 2012, Alvina was awarded the Vavilov Medal in recognition of her contribution to the conservation of plant genetic resource collection. Her professional affiliations include membership in the Council of the International Society for Horticultural Science. I'm sorry, but the long words are rather difficult to pronounce. And the IUCN Species Survival Commission. She also serves as the national coordinator for the European Cooperative Program for Plant Genetic Resources and as the national focal point for EURISCO. Her extensive experience includes serving as an international expert in numerous projects implemented by the FAO, European Union, World Bank, and UNDP. As a personal note, I visited this year Alvina's centers in Yerevan, and I must tell you that they really do impressive work. Her talk today will be on the early steps and emerging results in the introduction of multifunctional plant varieties in Armenia. So Alvina, if you are online, please.
Thank you, Chair. Let me share my presentation. So I'm happy to share with you early findings and perspectives of the introduction of assessment of multifunctional plants in our country. Let me go ahead. This presentation is based on a project titled Assessment of the Functional Value of Multifunctional Plants and Promote of Diversification. Through this initiative, we aim to explore the potential of multifunctional plants, but to support the implemented by the Scientific Center of Vegetable and Industrial Crops of the Ministry of Economy of the Republic of Armenia. The project is implementing in partnership with our foreign partner, Dr. Radu Verascu from the National Institute for Research and Development in Chemistry and Petrochemistry in Romania. Using this opportunity, I would like sincerely thank Dr. Verascu for his valuable support and willingness to cooperate with us on a voluntary basis. We first learned about him and his research through published scientific papers, and his advice was very helpful in developing project proposal, which was successfully selected with high evaluation scores. The project duration is 3 years, starting from 2025 till 2027. And the project is financially supported by the Scientific Committee of the Republic of Armenia. What is the aim of the project and the rationale behind? The aim of the project to expand the species and varietal diversity of multifunctional plants in Armenia and to promote their use in functional food and beekeeping. We are focusing on plants that combine multifunctions, where serve as the honey-producing plants, the medicinal plants having aromatic spicy properties. And this is our goal, to promote their use both in functional food applications and in beekeeping. The rationale of the project is closely linked to climate change, which already impacts on many sectors of Armenian economy, particularly agriculture and biodiversity. Numerous studies conducted in the country on developing of climate change scenarios and on assessment of the adverse impact of climate change show vertical shift of the existing boundaries of main natural ecosystems upwards by 250 till 300 meters, the reduction of the area of alpine and subalpine zones by 21-22%. Therefore, it is obvious that this ecological shift alerts the life cycle of the plants, duration of flowering and growing, and consequently the interactions with pollinator. At these conditions, one of the most practical adaptation measures is the diversification of agricultural systems. And by expanding the range of multifunctional plants and integrating them into existing agricultural practices, we aim to build systems that are more sustainable, more resilient, and productive under the climate change conditions. The three objectives were defined to achieve the goal of the project. The first objective is to investigate the bioactive compounds of various varieties of multifunctional plants in order to identify varieties which are most suitable for use in functional food. The second objective is to evaluate the adaptability of multifunctional plant varieties to our local climatic and soil conditions by using a combination of both biological and agronomic indicators. And the third objective is to access the melliferous potential of the plant varieties and recommend the cultivation of the more promising ones as honey-producing plants. Which plants species were selected for our study? We select the following three plant species: Moldavian dragonhead, Dragocephala moldavica, hyssop, which is Hyssopus officinalis, and edible chrysanthemum, which Glebeonis coronaria, which is synonym to Chrysanthemum coronarium. It is worth to mention that out of these three species, two species, namely Moldavian dragonhead and hyssop, are already known to grow in Armenia. In particular, Dragocephalum moldavicum is observed in Yerevan floristic province in dry landscapes, and Hyssop officinalis species is recorded in foreign floristic areas, so it's more widely distributed, also in dry and stony habitats. But there's no selection and cultivation activities carried now so far in the country for these two species. And the fact that these two species in the wild already can be found in the country served for us as a sign of good potential to adapt to local climatic and soil conditions. And other factor which we also consider while select the plants for our research is that these three species, according to the literature data, are relatively tolerant to varying soil and climatic conditions. So we can suppose that they are suitable for cultivation in farms which are doing green, which are accepting principles and approaches of green agriculture and organic farming. By focusing on these plants, we aim to explore both their functional food value and their role in the sustainable agricultural systems. We start our project with some preliminary studies. Which studies were about. So we reviewed existing ex situ collections and databases. Since we are at first time working with these species, we needed to understand the availability of relevant plant varieties, what exist in other countries, which varieties are developed, what type of germplasm is conserved in the national gene banks. And we did the following analysis. You can see on the screen that we analyzed distribution of Hyssop officinalis accessions per national inventories. We reviewed distribution per origin, later analyzed accession number per state select categories: wild, weedy, traditional land races, breeding research material, advanced on the approved cultivars. And also we analyze it, number of accessions included and multilateral system of the International Treaty of Plant Genetic Resources. For this analysis, we reviewed national databases of many countries, including Czech Republic, Hungary, Romania, Poland, Ukraine, France, Slovakia, Spain, and others. And we also reviewed regional databases like Mediterranean Gen Plus database, Baltic Gene Bank Information System, EURISCO, which is the European Catalog of ex situ collections. And also we reviewed and analyzed it, the availability of accessions of species of our interest in global international platforms like Genesis and GBIF, global facility of global biodiversity information facilities. The same analysis were done for Dragacephalomoldavica accessions, again across national inventories, distribution accessions per origin, and by the status of recorded ex situ accessions. The same categories were used. What are the results of these preliminary studies? So we found by overall that ex situ collections are relatively small. I mean ex situ collections of these three species selected. The lack of the names for many accessions complicated the selection, and large number of accessions are documented in databases from Ukraine, Bulgaria, Hungary, Romania, and we also found by distribution pattern Romanian and Ukrainian origin, and majority of M belongs to the wild species category. Not all accessions were recorded with the corresponding accessions name, so it was difficult to us to understand what's the name of variety and what's the name of the species. And category advanced or improved cultivar, which is crucial for our research, includes very limited number of accessions, only 24 accessions of hyssop and 12 of Moldavian dragonhead. And as for the MLS status, and this is based on this we can say that the accessibility is quite limited. Having this in mind, we choose to get seeds from the seed market, and we selected varieties which were readily available at the time. Some words about the experimental site and growing conditions. Experimental farm is located in Darakert community of Ararat province of Armenia, which is located in Ararat Valley, in the heart of Ararat Valley, which is the most cultivated arable land, with the quite warm winter, up to minus 2.6 degrees of Celsius in January, and very hot summers, in average 26.2 degrees of Celsius. Annual precipitations are small, about 289 millimeters in average. We cultivate our species using seedling production, seedling sown at the end of March, insemination chamber used for initial growth to ensure a high germination rate, and seedlings were grown under greenhouse condition before they were transplanted in the mid of May in open field. And the planting scheme was designed with 60 centimeters between rows and 35 centimeters between plants to ensure optimal space for growth and evaluation of plant performance. I have to say that we decided to grow the plants through seedlings rather than direct sowing. The main reason for that decision is that hyssop is a perennial plant, and when sown directly from seeds, it usually does not flower and produce seeds in the first year. And by using seedlings, the plants are able to flower and set seeds during the first growing season. And other advantages of seedling production includes ensuring uniform planting material and providing plants with a well-developed root system, which supports better establishment of the shoot height, number of lateral shoot, length and width of the leaf blade, height of corolla in Florence, width of flower lip, lower lip of the flower. And as for biochemical analysis, we determine total phenolic content, total flavonoid content, and phenolic acid contents. You can see on the screen the methodologies used for this biochemical analysis. I will not go into details to save the time. Some words about the phenological observation. You see on the screen a phenological observation on hyssop example. The most valuable parameter is the flowering stage duration. We calculate it in days. And conclusions we can get that all varieties of the three species can successfully completed all developmental stage and produced fully mature seeds. Some varieties exhibited a slightly longer growing season by about 2 to 3 days compared with other varieties. And these include Tai Shiban from edible chrysanthemum, Moldavian which for Moldavian dragonhead, and Lecar Ine and Accord for hyssop. These observations are important because they provide insights into the growth dynamics and maturation during maturation timing for each individual variety, which can influence functional food applications and agroecological integration. On this slide, you can see the pictures which we use to make our morphological analysis. As I mentioned before, we measure the shoots, a number of lateral shoots, we measure the flowers and leaves. Here's an analysis of varieties of Hyssopus officinalis. And the next slide, Dragocephalum moldavica varieties, and this is for edible chrysanthemum. Some results of this morphological analysis you can see on these two tables. The first table presents plant morphological parameters of the different varieties of Dragocephalum moldavica, and the second table presents flower morphological parameters also of Dragocephalum moldavica. Conclusions are following. The analysis of morphological parameters show it by under the local condition of Ararat Valley, the difference between varieties of all studied plants are minimal. They are exhibited similar overall growth habits. However, some differences were observed in specific traits. In particular, Garinage and Gorgona were characterized with the tallest inflorescence, while Amethyst had the largest corolla and widest lower lip of the flower. These characters indicate potential advantages for pollinator attraction and for use in ornamental purposes. In contrast, Sultan and Moldavia showed more compact growth, which may be preferable for high-density cultivation or used in the regions with quite strong winds. These observations help guide the selection of varieties for different cultivation purposes and ecological conditions. Now, some words about agronomic indicators. We use two parameters: green mass yield, expressed in kilograms per square meters, and weight of 1,000 seeds in gram. So, Regarding green mass yield, the highest biomass was observed at INI, variety of Hisopus officinalis, while the lower was Lazur, and the Leca varieties, Lecker and Accord, had intermediate value 0.51, 0.48. As for 1,000 seed weight, it was very similar. Picture was very similar across all varieties, ranging from 1.19 till 1.21, which is recorded for any variety. This suggests why any varieties is the most productive in terms of both biomass and seed potential, and seed size is uniform across varieties. All of them are suitable for seed production. Now let's go to the biochemical analysis. On a screen, you can see the diagrams illustrating total phenolic content in different varieties of Hisopus officinalis. A second diagram illustrates total flavonoid content in Hisopus officinalis varieties. And on the next slide, you can see the diagram illustrating total phenolic acid content, again, in Hisopus officinalis varieties. What are the general findings? The cultivars demonstrated notable difference in total phenolic content, total flavonoid content, as well as phenolic acid levels. This research emphasized the potential of Hisopus officinalis varieties as a rich source of biologically active compounds, which are used and is suitable for functional food applications. Now about some specific result. Let's go now look on a biochemical profile of the three Hisopus varieties. Starting with the total phenolic content, we observed variation among varieties. Accord exhibited the highest TPC, followed by Lazur and INI varieties. As for you know that the phenolic content compounds are well known for their antioxidant properties, which contribute to functional foods. And the highest total phenolic content in Accord suggests a greater capacity to neutralize free radicals and to reduce antioxidant stress. A similar pattern was observed for total flavonoid content. Again, Accord variety had the higher value, where INE and Lazur were slightly lower. And since flavonoids play important role in antioxidant and anti-inflammatory and antimicrobial activities, this indicates that these varieties, especially Accord, are promising for functional food developments. And finally, for phenolic acid, Accord showed again the highest content, followed closely, very closely by INE and Lazur. Overall, Accord shows consistently the highest level of biologically active compounds investigated, and high phenolic and flavonoid content indicates strong antioxidant activity, antimicrobial potential, and this highlights suitability for functional foods. On this screen, you can see the table summarizing morphological highlights and biochemical profiles. And using these results, we can come to the conclusion that all varieties of Hisop completed growth stage and produced seed. Morphological difference in Hisop varieties were minimal. Notable traits observed in Accord, INE, and Lazur. Biochemical analysis illustrates that Accord showed the highest content of total flavonoid and total phenolic compounds, phenolic acid as well. Accord, so based on this, we can say that accord variety is the top performer for biochemical activity. So it's ideal for functional foods and for pollinator attraction. Any variety combines moderate biochemical content with the compact grow, making it suitable for high-density cultivation. And for lazur variety, we can make the conclusion that this variety shows a balanced biochemical and morphological profile, promising for suitable sustainable agriculture and ornamental applications. As for Moldavian Dragontain varieties and edible chrysanthemum, phenological observations also show that all phenological stages passed successfully, and plants, all varieties produced seeds, so they are well adapted and can be introduced in the country, and morphological and biochemical assessments are still in progress. So overall insights, yearly results confirm the adaptability and potential of multifunctional plants under the local conditions. Data highlight promising varieties for functional foods, beekeeping, and sustainable cultivation. Looking ahead, we are planning the next steps. In September this year, we had the pleasure to host Dr. Radu Verascu at our scientific center. During his visit, we had very productive discussions regarding the continuation of the project and future priorities of the research. One of the main upcoming activities is the analysis of the essential oil content in all varieties in order to understand composition and functional properties for functional food applications. And further morphological, agronomic, and biochemical evaluations will be done for selected promising varieties. It's all. Thank you very much.
Thank you very much, Alvina. As I've already told you, we visited the laboratory, but sorry, what I forgot to tell you is that we also had the chance to visit the country. It's a very nice country, and I recommend you all to visit Armenia. It's a direct flight right now, so it's easy to go. So if back to the presentation, if we have any questions from the on-site participants, please. Okay, or from the online. I don't see any hands. In that case, Alvina, thank you once again for your presentation, and we will be waiting for you next year here at ICECHIM to continue the work. Thank you.
Thank you very much.
We will now go to our next guest, Dr. Martin Kilo, from the Fraunhofer Institute for Silicate Research in Germany. Dr. Martin Kilo is a senior scientist at the Fraunhofer Institute for Silicate Research, Brombach branch, Germany, where he leads research activities in glass conservation and material science. He holds a doctorate in chemistry from the University of Bayreuth, where he studied heterogeneous catalytic reduction of CO and CO2 under the supervision of professors Vaucaun and Biker. His career includes position at the Technical University of Clausthal, the Fraunhofer in Würzburg, and a temporary professorship in glass technology at Technical University Bergakademie Freiberg, where he also served as a Dean of Studies. Martin's experience spans glass technology, heterogeneous catalysis, and mineral materials, with significant contribution to the conservation of glass heritage and the development of sustainable materials. Sorry. He has conducted research stays at leading institutions, including École des Mines Nancy, Keele University, the University of Cambridge, and the Indian Institute of Science in Bangalore. He is chair of HVG DGG Glass Forum, expert in chemical analysis of glass wool, and co-founder of Glass Campus Torgau. He was also actively involved in organizing international events during the United Nations International Year of Glass 2022. And besides all this very rich CV and very many words, Martin is a very good collaborator. It is at the core of consortium that right now it's I think it's the third attempt in a Horizon call. He was so kind to agree to participate with us in many proposals, and I think that we can call him up to now a very good friend of our institute and one of my very close collaborators in Europe. So Martin, please.
Thank you, Rado, for the introduction. Can you hear me and can you see my presentation?
Yes.
Okay, so then welcome to my talk. It's not given by me, it's given by me, but the largest part of the work has been done by Mrs. Maas-Diekler, Mrs. Rother, and Mrs. Leisner. And we are from the Fraunhofer ISC, and I'm located in an old cloister, so in a very nice location in the middle of Germany at that time. Yeah. So the talk is about the glass dosimeter. You can see here two examples in church windows, one at the surface and the second on the top left, which is between the glass and another protective glazing. And so to start on, what are glass dosimeter and what are they used for? They are used for glass dosimeter are integral dosimeter, so where we monitor the atmosphere over a longer time scale can be from months to year, and to see what happens when what happens with the microclimate at this position. In order to develop this dosimeter, we have developed different types of corrosion-sensitive glass, and the corrosion can be due to several environmental impacts like temperature, moisture, but also reactive gases. Also, microorganisms can lead to the corrosion. And as I mentioned, since we have different glass compositions, we have also dosimeters which are sensitive for different sensitivities, both different gases, but also different location. And how to make them? We made a cut of a block of glass. We cut it in pieces, in slides. Then we put them, we polish them. Then we put them between dia framing, an old dia framing. Maybe it's older if you know the dia system. So they are a few centimeter, and then this dia with the dosimeter in the middle can be placed more or less wherever you want in any position. So what happens then with the dosimeter after some exposure? So you can see here on the left shows how it looks when it's fresh, and on the right how it looks after 3 months of exposure in a church in Deckenfrom, which is a small town somewhere in Germany. And in the middle, you see what all can happen during this time. There can be some moisture, there can be some gases which are reactive, there can be also traffic and some other surroundings in the town or in the large town, or which can affect this dosimeter and which makes them these corrosion products. And what are the typical corrosion reactions? We have normally the normal glass, and then during the reaction there is an exchange of protons with alkaline earth ions from the glass, and there can be even reactions to form the corrosion layer, to form a corrosion product layer, but also on top on an alkali leached water containing tea layer on the surface of the still pristine glass. And the idea is taken from the corrosion which can be observed, for example, in medieval glass, where there is a sodium, where sometimes there is a potassium-rich glasses, and then there is some glass corrosion in this glasses observed. And at the end, we have these corrosion reactions. And what we do for the glass evaluation, I hope you can hear me because we have some work doing. I hope
yes,
not too loud now in my background. I'm sorry for this, but some people we get some new windows there. So what we can see is when we make an infrared spectrum that in the water range at around 3000 centimeter, that initially we have only a very flat band. After some time we have, or after reaction, we have a very strong band. So in the left side it is in the pristine sample, and the right side from the corroded samples. So what we make for data evaluation, we make typically an IR measurement in the transmission mode of the water and the OH band, which comes in this region together. What we can see here is sometimes the formation of corrosion crystals, which are not so critical, then the shield formation, but also sometimes cracks, what happens after the exposure. And then we extract the so-called the delta E value by measuring the difference between the initial here in blue and the final signal. And the larger the increase of this band at the 3300 centimeter is, or at the reciprocal centimeter, the larger the corrosion is. And as I mentioned, we have several different glass compositions. In some glass compositions for the sensors, the effect is large, and in others, the effect is not so large. So this is one measurement result. The other is really with microscopy shown here to investigate the type of corrosion crystal forms, but I come to this later. So then just where can these glass dosimeters be placed? They can be placed in display cases or in the deposits in museums. They can be placed in somewhere in buildings to monitor building conditions. So it can be both in the outside, but also in the inside. We have used it, as I showed in the initial slide, for evaluation of the protective glazings in stained glass windows in church typically, but also in some other buildings. And here, there also it can be both in the inside as well as in the outside. And finally, we have also used it for monitoring some industrial sites to see how this the atmosphere of industrial sites in some foreign countries will affect the production. This comes from some companies who goes from the production to some foreign and cheap countries, typically in Malaysia or something, countries like this, or China, where it is sometimes the climate is much different from Europe. So that the companies also want to know what happens when they go to a foreign country, in particularly if they have processes which are very moisture sensitive, like, for example, painting some cars or so, when a car pass, where some car companies had wanted to make an investigation of this. So just here, some what happens when we place dosimeters on the top left in the cathedral in Cologne. In the top left, there are here is a view from the outside of the Cologne Cathedral. The cathedral had a protective glazing, and what we have placed all the time, 2 different glass sensors with different reactivities and selectivities. And they have placed them, we have placed them in 3 positions: the fully outside position, the middle position between the protective glazing and the original window, and the inside position in the bottom. It is a view from the inside from the church, where the 2 inside glass sensors are shown. And then they were kept there for half a year. And then you can see the P1, P2, and P3 means the different positions. Sometimes we have here the P4, which are the inside at different locations. And you can see what is here. So SX, 8E, and so this is labels for a particular window and a particular part of the window. You can see depending on the position in the cathedral, you can have a large effect, but also not so large effects, which means we have placed them also in the close to the ground floor, but also in the top part. The top part is more prone to atmospheric, so more rain, more wind, and so we come to the top part. But also in the bottom part, it is inside the church, then there is more where the peoples are walking and visiting the church, while in the top part of the church inside, the effect of the peoples when they inhalate and exhalate is a little bit less. It is more, I would say, exchanged. So you can see that at a different location we have different corrosion effects, both in the inside and as well as in the outside. Which means that even within one church, which is a large cathedral, you have different microclimates at different positions in the church. And also in the Rosetta, which is a large really large part, you can have a quite hard position. Also, it's depending, which I haven't mentioned, if it is from the orientation of the cathedral, if you go the western side where there's more rain, or if you go to the eastern side where normally less rain is coming, because mainly the rain is coming from the west and not from the east, you can see different effects. Also, this is a microscopic evaluation of the glass dosimeter. You can see that in the position between between the 2 glazing, the effect is much less than in the outside position. And so this can be also used to monitor the quality of this climatization system between the 2 glazings, because if the climatization between the outside, the protective glazing, and the original historic windows is not well, then sometimes the corrosion between these 2 glazings is even worse than outside. So sometimes the glazing is not well if there's no climatization going up in the system. So here's an example of the climate control in museums, where and why and where. Here it is a so-called Royal Household in the Museum Neues Grüngewölbe in Dresden in Germany. It is from the 18th or 17th century, a large exposition with plenty of enameled figures, and also which was placed in a display showcase. And they built after the reunification 91, they have a new showcase. But after several years, in the year 1997, it was observed that there is in this enamel figures some parts of the enamel was falling down, and pieces of the enamel were falling down, and some corrosion, which means that there's a corrosion. So after 20 years, we have been working with them for several years. And after 20 years, they have even produced a new showcase. So we have helped them to optimize the showcase by doing several things like removal of the press board, which was part of the showcase, which had a very strong effect. Then they have put some activated charcoal filter in it, having an effect. And they had a better ventilation system, and even with green one, even more powerful ventilation system. But still there was some corrosion, so there were 4 campaigns in the years 97 to 99. So at the end, at the year 2017 or so, they placed everything on a new place. There was a completely new display concept, including new showcases and so on. And here there were really almost no corrosion, and so there's a real, this system, very optimal condition. And meanwhile, we have also developed this museum concept to restore the damaged enamel objects, because there were plenty of objects which were damaged. And in this new showcase here location, now the restored and perfect, again perfect objects are on exposition. You can also have a problem in some in some storage, because normally in a museum 95% of the objects are stored, but only 5% or something like this are really for display. And in the storage, sometimes the conditions can be very different from the condition in the museum itself. In the depots, they have sometimes much different conditions. And also in the depot, you see they have often expositions or some systems which are not perfectly like shown here. Which have, in the case of glass, can have different glass diseases. The sample stores them more or less forever. The museum stores them more or less forever. But they are in the storage, and they can have different types of glass diseases, which are glass corrosion. So some object from the museum in also with more or less severe glass corrosion effects, craquelé formation, and things like this. Also both in old objects from the 18th century, but also in newer objects from the early 20th century. Also here, there is here the glass corrosion can happen, and the glass corrosion has led to this corrosion of the objects, and therefore the formation of glass diseases, and sometimes there are even tears or droplets on the surface of glasses which look like tears. So the glass, in particular, if it is corrosion-sensitive glass containing a large amount of potassium, can even have some droplet formation. Can be shown here for watery glass, where then is a really droplet of formation. And these droplets are highly acidic. It's like, so it is it is basically sodium or potassium hydroxide. And therefore the droplets stay liquid, and you can even see it in an image series when you see here. And initially you have some in the microscope. Initially there were some crystals, but after some times there's really a formation of droplets, and since they have a very high pOH, there's also this liquid remains stable, so it remains stable for long times, and therefore this is then if you then clean it, then you will remove part of the glass, or if you then wipe the liquid away, then part of the glass will be removed. Then you can so also you can have see that glass diseases can be also depending on the structure. Shown here is a glass piece where the decoration was cut out, and then they have different glass crackle. Therefore, the rough glass surface leads to different corrosions than the pristine surface of the object. And even what you can see here, when you look on the right side, you can see sometimes that the crackle formation is different. Here you have some plates on the top, and on the bottom you have more vertical cracks. So in the top you have parallel to the surface cracks, on the bottom vertical to the surface. So this is due to the different production of the glass in the time when it was produced. So this means that glass dosimeters are very helpful in the museum, both in the glass showcase as well as in the storage. In storage, it is also particular to avoid further corrosion, because if you then dry the object too harsh, then it could be really detrimental, and the corrosion can be even stronger, and these platelets can fall off if the atmospheric condition is too dry. So the moisture will even help to protect, to keep these platelets at the surface. And so here, therefore, even in the store case, you have to monitor the atmosphere. You should monitor it. So we have also what I just at the end very short view on the material developed. We have also developed some coating and consolidation material for glasses, glass glazings, enamels, metals, and so on. And the people from this museum in Dresden have used these materials to repair this old enameled objects. And you can see this system what we have developed is very long time stable. We have shown that it is really for even 50, 60 years of storage. It's fully workable, and there's also no yellowing effect, which is by conventional systems very often observed. And here you can see what happens with one of the objects from the systems after some times. We have also looked what happens if the surface is curved and not flat, and also if there are different colored enamels. And what can be seen here is that these that here in this part there was a strongly enameled damaged enamel. It was then restored and even showed almost similar after 12 years after the restoration. So there were nothing, no effect. So what is in the left build image is so brownish, was really fallen away, and it is now stable. So to come to an end, these glass dosimeters help to monitor the corrosive microclimate in the cultural heritage. And it can be, for example, in the cathedral, as I mentioned, in the top of cathedral, different from the bottom, or on the western parts, different from the eastern part, and so on. It is an integral sensor system, and it can be used outdoor, indoor, in display cases or showcases, or in some affluent location. Also in cathedral, we have used it in some cryptas and some special rooms. What we want to make in the future an automated data evaluation, because at the moment we still have to place them there for months or years, then take it away, measure them out home in our labs, and this is quite time consuming and not so easy. So we want to use an fully automated data evaluation direct on site, And we also want to have in the future more class composition with some larger and broader selectivity. And also this is in particular helpful when, because when for this global climate change monitoring to see what happens with the cathedral, when the summer is getting hotter and drier, or the winters will get more wet, or more intense raining effects will happen and things like this. And with this, thank you for your attention. Thank you.
Thank you very much, Martin. Your work in the cultural heritage field is truly impressive. We are well familiarized with your work. So congratulations from my side. And if we have any questions from the participants, please, or from the online guests. If not, I do hope, Martin, that we will get to implement together those future perspectives that you put on your presentation. I am sure that
thank you for organizing this very excellent conference or symposium.
Finally have that project that we are talking about.
Yeah.
Thank you once again.
Okay, thank you.
And now I think we can make a 10 minutes break and return with the oral presentation section in around 3 and 10 minutes. Thank you.
And the first work is presented by our colleague Andrea Luisa Merz. She is online because she is performing on research stage abroad. And the title of her presentation is Synthesis of Dendritic Bioactive Glass Nanoparticles and Their Application in 3D Printed Hydrogels for Bone Tissue Engineering. So Luisa, if you are online and you are ready for presentation, you have the floor.
Yes, thank you. Can you hear me? Thank you for the presentation. So yes, I'm Luisa Amert, and today I will talk about a part of my PhD thesis about the synthesis of dendritic bioactive glass and their application in 3D printed hydrogels. As life expectancy increases, bone-related diseases and fractures are becoming more common. And by 2050, 4.5 million people will be affected by bone fractures. And even now, over 200 million individuals suffer from osteoporosis. So conventional bone grafting methods can keep up with the increasing demand of it. That's why bone tissue engineering has growing fastly in the last couple of years as an alternative, as a promising alternative. And bioactive glass is one of these biomaterials with which are fascinating because they can release some therapeutic ions that can stimulate either angiogenesis or osteogenesis depending on their composition. Usually they have a round shape with some cylindrical pores, but in this case I will focus on the dendritic shape, which is similar with tree branch and has this like a flower-like architecture seen in plants. The hydrogel used initially was alginate because it's a common one, biocompatible and non-toxic. But because it's unstable degradation profile, we tried to oxidate the alginate to modify the degradability. That's why we oxidize it with sodium peroxide to obtain these aldehyde groups that were further reacting with the amine group from the alginate, gelatin, sorry. And that's how we obtained oxidized alginate gelatin, shortly Adagel. To synthesize the nanoparticles, we used two surfactants that were dissolved in water and to form the micelles, and Theos was added as a silica precursor. The powder were dried and calcinated That's how we obtained the first powders, the DMSN, dendritic mesoporous silica nanoparticles. But because we wanted also to add some calcium inside, we add these particles into a calcium aqueous solutions. We let it to steer for 1 hour, and the powder were dried and overnight and calcinated at 700 Celsius degrees. That's how we obtained the dendritic mesoporous bioactive class nanoparticles, DMBGN. 0.1 of these nanoparticles were added in a 5% ADA solution. It was stirred and ultrasonicated, the solution, and after we added a 7.5 gelatin solution, and that's how we obtain our ADA gel with nanoparticles hydrogel. The hydrogel was further used for printing, so we placed it into a cartridge, stored it in a fridge for a couple of minutes, and then we used an extrusion-based bioprinter to form these scaffolds. Before using the scaffolds, we did an additional cross-linking. To check the printability of our scaffold, we performed 3 different tests to check the reproducibility of our 2 types of hydrogels. Talking about the results, by SEM we can see the shape of our particles, cylindrical and with a diameter around 80-85 nanometers. The EDX showed the incorporation of our calcium inside, around 6% of a molar composition. The zeta potentials show the negative surface charge due to the silanol groups on the surface with a monodispersed distribution. The adsorption and desorption isotherms showed type 4 isotherm with an H-free hysteresis loop, and by BT analysis we saw that our DMBGN has a lower surface area than DMSN, most likely due to the fact that calcium somehow acted as a network modifier in our silica network. Further, we did some FTIR and XRT on our nanoparticles. The FTIR showed a free common band in a 4 silica network, and the XRT showed around the 22 degrees diamorphous peak that we wanted to see. Okay. After with both of our nanoparticles, we immerse them in SBF, in simulated body fluid. And as we can see in the SEM images, after 3 days, 7 days, and 14 days, in the figure A, we can see after 3 days that there are some needle-shaped-like structures which show the hydroxyapatite formation already after 3 days. And in the figure C, after 14 days, we can basically really see the hydroxyapatite formation, and the FTIR is confirming the same thing. Around the 569 wavelength, we can already see the phosphorus oxygen vibration in DMBGN, while with red is the DMSN FTIR, and we can see the hydroxyapatite bands just in the case of 14 days. And the XRT is confirming exactly what I already said about the FTIR. And the results about the ADA gel, about the scaffolds with nanoparticles. Here I will talk just about the ADA gel with the DMBGN. We can see the porous structure in the SEM images. The XRT is showing no crystallinity happened during the experiments. And the FTIR showed that we have these carbon-nitrogen bonds around 1,600 and 1,500, which confirm that our oxidized alginate reacted with the gelatin. And for the printability tests, we can see in the figure C, where we have the ADA gel DMBGN, we can see the grid structure. It's better than in the case of a simple ADA gel taken as a standard, or the ADA gel with DMSN, most likely due to the addition of the calcium inside. The diagonal crossing ratio and the printability factor graph are showing the same. The ADA-gel DMBGN have a better stability than the DMSN ones. And talking about the strut width, we can see in the pictures below that if we are taking ADA-gel as a standard, the printability went well, but seeing the ADA-gel DMBGN, we can see some disruptive in the ink, which is most likely due to the fact that the ADA-gel DMBGN's is crosslink is gelating faster than we expected. And in conclusion, we obtained some dendritic uniform nanoparticles of dendritic bioactive class, which shown some good in vitro bioactivity, especially the DMBGN's ones. The printability went well, showing that the ADA-gel DMBGN is a promising ink for scaffolds. But for the rheological characterizations, it's needed to see exactly the effect on bioprinting of adding this nanoparticle inside of our hydrogel. And if we want to add more ions, another elemental composition technique is needed. And thank you for listening.
Thank you very much, Luisa. If there are questions from the conference hall or from online. Okay, no questions. Good luck, Luisa, and let's hope that you will finish as soon as possible your PhD thesis, and we wait for you here at home.
Thank you so much.
Thank you. So we pass to the next presentation made by Olena Ardușenko. She's coming from Brazil and she is here in the conference room. She will present the paper, the work entitled Purification Strategies for Biocompatible NP-doped carbon dots from Synthesis to Safe Medical Use. Olena, please.
Hello, everyone. My name is Olena Ardușenko. So I'm presenting now Brazil, Pontifical Catholic University of Brazil, but I'm originally from Ukraine. And now I'm doing postdoctorate there in Brazil. And this research was conducted in Brazil. So the title of my research that I'm presenting today is Purification Strategies for Biocompatible Carbon Dots from Synthesis to Safe Medical Use. Let's start with a quick introduction. Carbon dots, I hope there are some people here who knows and who works with carbon dots. Carbon dots, they were discovered in 2004. And since then, they have become the hottest topic of materials researchers. And it's easy to say why, because they have nanometric size, low toxicity, high photostability, high surface area, tunable photoluminescence, and also simple scalable synthesis from inexpensive precursors. And that's why they're widely using now in bioimaging, drug delivery, photocatalysis, electrocatalysis, sensing, and so on. However, synthesis of carbon dots is just half of a battle. When we synthesize carbon dots, we also have molecular fluorophores, they byproducts during the synthesis. And sometimes there are some articles that these byproducts, they can be almost make up almost entire sample, up to 98%, with only 2% of what we think carbon dots. And this leads that to we can imagine that two research groups working of what they think the same material, but reporting completely different results. One group can report green emission of these dots, another group can report blue emission, and simply because their purification strategies were not the same. And they're working, actually they're working with the different mixtures of carbon dots and molecular fluorophores. So this lack of reproducibility is now recognized as one of the main bottlenecks in the carbon dot field. And that's why purification is not for carbon dots is not optional. It's a critical step that must be taken before we can trust the data or move forward to biomedical application if we want to have reliable and reproducible results, reliable and reproducible chemical properties, optical properties, and if we want to use these carbon dots in bioimaging. So let's see how people usually purify carbon dots. The common methods are filtration and centrifugation, dialysis, sucrose gradient centrifugation, exclusion chromatography, electrophoresis, and so on. But filtration and centrifugation can remove large particles, but not molecules. Dialysis is cheap and simple, but it's extremely slow and often incomplete. And other methods can be effective, but they require complex steps and hard to scale up. For example, let's take dialysis. Dialysis is the method when you put your carbon dots in a semi-permeable membrane and you run water. You run water during days, sometimes weeks. And of course, after this method, you can remove salts and some reactants, but this method does not completely remove molecular fluorophores and also may fail if the byproducts are poorly soluble in water. And the other problem is that most of the researchers, they use dialysis membranes with a cutoff around 5 kilodaltons. There is an article about this. They calculated how how people like purify carbon dots, but they also calculated that the ideal size of this membrane should be around 20-40 kilodaltons and not 5 and not 1. It's not enough. So people working with not pure material. So to address this problem, we synthesize crude material, crude carbon dots, and nitrogen and phosphorus doped carbon dots, and then we use two purification strategies, dialysis and chromatography. We compare these strategies. We also characterize our material, our crude material, with different methods, and we show that our material is have good biocompatibility and can be used for bioimaging, but our material is not clean and need to be cleaned before the use. So, and our two purification strategies, dialysis. For dialysis, we used a membrane with molecular cutoff around 12 kilodaltons, and the process was conducted in continuous water flow for 3 weeks. And for column chromatography, we used like purification with on chromatography column with further Soxhlet extraction. We packed silica gel column and eluted the cruise suspension using water. Then we recovered this silica with the remaining adsorbed dots in the silica, and we applied Soxhlet extraction with boiling water for 24 hours. However, there are two problems. One of the synthesis and this formation of these byproducts, molecular byproducts, and another problem is validation of purification process, because the common methods used for analysis of carbon dots, like microscopy, TEM, and IFM microscopy, they cannot distinguish molecular aggregates with really carbon dots. So common methods like microscopy is not enough. We need to have something else. And this something else is, so there is an article of these authors, Bartolome and co-authors, published in 2022, and they propose to use proton nuclear magnetic resonance for this. Because in the spectra of nuclear magnetic resonance, when we have sharp narrow peaks, it's indicate that we still have molecules in the sample. And if we receive these broad features, broad peaks, it's what we expected from the true nanostructures. So we can use this to validate our purification process. And in our research, we also adopted this method. Just to remind, we synthesize crude product, characterize it, and then let's see what we have in proton nuclear magnetic resonance. For raw material, we received sharp peaks, what is characteristic of the presence of molecules. So after dialysis, we have less peaks, but still many remain. And after purification by column chromatography with Soxhlet extraction, we received finally broad features, and sharp signals are gone. We also used IFM microscopy just to confirm and the same materials. So in IFM of raw material, we have a lot of impurities full of aggregates. After purified by dialysis, some impurities are removed, but the surface is still heterogeneous. And after chromatography, we received well-dispersed material with narrow height distribution. And purification is not just only about the process, as it's also about sustainability. So we compare two methods, our method and dialysis, through three scales: EcoScale, AgriPrep, and RGB12 metrics. In all three cases, chromatography performed better. It was faster, greener, and more efficient. This makes the chromatography the best choice for purification of carbon dots for biomedical applications, where both safety and sustainability are important. So none of this work would be possible without my amazing team from a laboratory of adsorbents of chemical analysis, environmental protection, and biomedical applications, leading by Volodymyr Zaitsev. He's Ukrainian and he's in Brazil. We also have another postdoc girl from Ukraine there, other group Brazilians, Colombian from Colombia. So you can join us on Instagram using this QR code. We post there our research, our opportunities. If you want to come to Brazil, we are open. Thank you for your attention. Thank you for finding agencies. And I want to leave this cartoon. And don't forget to clean your carbon dots. It's funny, but the message is serious. If we want reliable, reproducible, and safe carbon dots, we must purify them and validate that purification process. Thank you.
Thank you very much for this nice presentation. And now, if you have questions from the room or from online, no. So thank you again, Elena, and good luck in your work and in your research. Thank you. And now I pass the microphone to.
And right now we are going to the next presentation. Is from Aminu Baya Muhammad. It's online. I'm waiting to enter from online. I am not so fat like, so you don't need to be scared. You need, yes? The presentation is a thermocatalytic valorization of waste plastic into fuels over bimetallic nickel and cobalt oxide on aluminum. It's online. It's about, okay. If it's not, we will go further to presentation from our partner from Anca Daniela Raicu. Sorry, because it's bolded the latest one. Sorry, it's the presentation from Anca Andreea Zoe, Nicoleta Moraru, Caterina Cristina Teodorescu Stănică, and Anca Daniela Raicu. The pharmacognostic aspect and phytotherapeutic application of Matricaria camonile, Melissa officinalis, and Hypericum perforatum, our dear aromatic and medicinal plant.
My name is Zoe Anca Andreea. I'm a student at Faculty of Pharmacy, University of Titu Băiorescu, and I will present pharmacognostic aspects and phytotherapeutic application of Matricaria chamomilla, Melissa officinalis, and Hypericum perforatum. Phytotherapy represents a significant branch of complementary and alternative medicine, relying on the use of bioactive compounds derived from medical plants for both preventive and therapeutic purposes. Unlike synthetic therapies, phytotherapy harnesses the synthetic effects of complex natural mixtures, resulting in immediate action and often improved tolerability. The growing global interest in natural remedies is driven by the need for safe, sustainable, and accessible therapeutic resources capable of addressing both acute and chronic health conditions. Pharmacognostic analysis plays a crucial role in the field, as it enables the correlation between morphological, anatomical, and phytochemical characteristics of plants, material, and their therapeutic potential. This interdisciplinary approach ensures the correct identification, authentication, and quality assessment of herbal drugs, with also providing valuable insights into the active constituents responsible for pharmacodynamic effects. Consequently, pharmacognostic studies from the Science Foundation for validating the traditional use of medical plants and facilitate their integration into modern medical practice. Within this context, Matricaria chamomilla chamomile, Melissa officinalis lemon balm, and Hypericum perforatum stand up as three plant species of major interest in contemporary pharmacotherapy. These species are widely distributed across Europe and are recognized for their diverse pharmacological properties, including anti-inflammatory, sedative, antidepressant, antimicrobial, and wound healing effects. A comparative study of their morphoanatomical and phytochemical features provide a comprehensive understanding of their therapeutic value, elucidating the relationship between structural, characteristical, chemical composition, and biological activity. Therefore, the present study aims to highlight the correlation between morphological and phytochemical profiles of these species and their therapeutic application, emphasizing the importance of integrating pharmacognostic data and experimentally and clinically demonstrated biological effects. Materials and methods. Comprehensive synthesis of pharmacognostic data and current clinical evidence was conducted regarding the chemical composition and therapeutic potential of 3 medical plant species, Matricaria chamomilla, Melissa officinalis, and Hypericum perforatum. The research involved a detailed review of science literature, including peer-reviewed articles, pharmacopoeias, and authoritative phytotherapy databases. Data collection focused on identifying and characterizing the principal bioactive constituents of each species, such as flavonoids, phenolic acids, terpenoids, and essential oils, and on evaluating their pharmacological mechanism and clinical applications. Emphasis was placed on study demonstrating their therapeutic relevance in areas such as anti-inflammatory, anxiolytic, antidepressant, and antioxidant efficacy. Sources were selected from major scientific PubMed, ScienceDirect, Fingerlink, and Google Scholar, prioritizing publication from the last 2 decades to ensure the inclusion of up-to-date findings. Results. Matricaria chamomilla is a medical plant well recognized for its rich phytochemical profile and broad pharmacological spectrum. Its volatile oil contains major constituents such as alpha-biasbol, chamazulin, and biasbol oxides, compounds responsible for its anti-inflammatory, antiseptic, and spasmolytic properties. This presence of flavonoids and coumarins further enhance its antioxidant and vasoprotective effects. Pharmacological studies have demonstrated that Matricaria chamomilla extracts moderate inflammatory mediators by inhibiting cyclooxygenase and lipogenase pathways, contributing to the attention of tissue edema and irritation. Clinically, chamomilla presentation are employed in the management of functional gastrointestinal disorders and in the cutaneous and mucosal inflammation, including dermatitis, gingivitis, and stomatitis. Its mild sedative and good healing effects justify inclusion in topical formulation and herbal teas used for relaxation and recovery. Melissa officinalis, lemon balm, exhibits a complex chemical composition dominated by volatile oil rich in citral, citronel, and linolol, alongside phenolic acids, especially rosmarine and caffeic acids, and flavonoids. These constituents confer the plant notable antioxidant, antiviral, and neurotropic effects. The antiviral activity against herpes simplex virus has been attributed to the synergistic action of rosmarinic acid and essential oil components. Consequently, lemon balm is indicated in the neurovegetative disorders, mild anxiety, sleep disturbance, and cutaneous viral infection such as herpes labialis. The plant safety profile supports its long-term use in phytotherapeutical formulation for stress-related and psychosomatic conditions. Hypericum perforatum is characterized by the presence of narphotodiatrons and fluoroglucinol derivatives and various flavonoids and phenolic acids. These bioactive compounds act synergically to produce antidepressant, anti-inflammatory, and antimicrobial effects. Clinical trials have validated as effective in mild to moderate depression, demonstrating efficacy comparable to standard selective serotonin reuptake inhibition, but with fewer adverse effects. However, is now to introduce cytochrome Phe450 enzyme and Phe glycoprotein, leading to significant herb-drug interaction when can reduce the plasma concentration of conventional medication, including antidepressant, anticoagulant, and oral contraceptives. Therefore, despite its high therapeutic value, clinical use requires careful monitoring to avoid pharmacokinetic interaction. Conclusion: the comparative pharmacognostic and phytotherapeutic analysis of Matricaria chamomilla, Melissa officinalis, and Hypericum perforatum underscores the relevance of collecting morphoanatomical, phytochemical, and pharmacological data with clinical evidence. Matricaria chamomilla demonstrates remarkable anti-inflammatory, spasmolytic, and digestive properties attributed to its essential oil constituents alpha-BS-bol and chamazulene and flavonoid content. Its efficacy in treating gastrointestinal and mucocutaneous disorders highlights in continued relevance as a safe and effective phytotherapeutic agent. Melissa officinalis is discussed by its neurotropical and antiviral potential, mediated by volatile oil components and phenolic compounds such as rosmarinic acid. Its sedative, anxiolytic, and antispasmodic effects, along with tropical antiviral action, makes valuable remedy for neurovegetative and psychosomatic imbalance, particularly those associated by stress-related somatic symptoms. Hypericum perforatum, through its content of hypericin, hyperophorin, and associated flavonoids, remains a cornerstone of phytotherapeutic management of mild to moderate depression. However, the potential of pharmacokinetic interaction necessitates careful monitoring in combined therapies. Collectively, these findings refine that the therapeutic potential of medical plants cannot be fully appreciated without the integration of pharmacodynamic characterization and clinical validation. The three species examined exemplify how phytotherapy, when scientifically substituted, represents a value adjuvant and conventional medicine offering safe, effective, and sustainable alternatives for maintaining health and treating disease within an integrative medical framework. And this is a selective relevance. Thank you.
Thank you. Questions? I have one or two. So the first one. For us, Hypericum perforatum, traditionally in the Romanian ethnopharmacology, let's say, Hypericum is known also for treating hepatical disease for its cholaretic and cholagogue action.
Yes.
Honestly, its antidepressant action was first of all for the American, because it's clear that they are the most stressed people of the world. So what it's showing more exactly the pharmacology. What is most relevant, the cholaretic, cholagogue, or the antidepressant activity?
I don't know.
Okay, so this is the this is one. This was the question. So, other questions?
I think that it depends of the how depressed is the patient. So it depends. So if there are, if there are no questions.
Yeah. No, it's because for several years and even now, the St. John wort, the Hypericum perforatum, is most used dietary supplement in the United States. Not for the hepatic diseases, for sure. Thank you. Thank you. Thank you very much.
Thank you very much. And we try again to see if Aminu Baya Muhammad is online and if he can present his work. Maybe if our colleague from IT can check if is this name online. In this case, we invite all of you to the poster session and downstairs, no, or here in for the to see a little bit also the equipments and then for the poster. Thank you very much.